So I don't need to explain to you how this functions because it's the same as the previous days. Since I didn't attend all the course of the previous days, don't hesitate to tell me if something you already know or is unclear in the chat or in the live document. Today I'll be speaking about gene function and gene expression and how they evolve in orthologs and paralogs. So I will have 3 parts. The first part I will be less about evolution directly and more about the bioinformatics of function and expression and which resource and database we can use, with the focus on the gene ontology and on the database Bgee, which we developed in my group for gene expression. And then we will speak about gene and genome duplication, and finally evolution after duplication. So first part, we're going to speak about gene function and the Bgee database. So this will be the more bioinformatic of the 3 parts, and the other 2 parts will be more evolutionary. And during this presentation, I'm going to try to get your feedback on various points. So for this, I ask you to follow this link, which is also in the live doc. And I will activate the first question. So now you will be able to answer normally poll about what is the function of the heart. So I don't see any answers yet, so I'll wait a bit. So you go to WooClap, the code is SIBCG3. And most of the votes are obviously to pump blood through the body. So in the case of the heart, this might seem obvious that the function of the heart is to pump blood and not to make a noise doctors can listen to. But if you think about when you speak about the function of a gene, if you were asked what is the function of BRCA1, for example, many people would say it's a breast cancer gene. But the breast cancer gene, saying that a gene is a breast cancer gene is a bit like saying the function of the heart is to get heart attacks. It's describing what it does when it doesn't do its job, right? So this makes us think more about, so this is your answer so far. Everyone is answering about pumping blood, obviously. But if now I go back to my slides, wrong thing. Sorry. Oh.
We are not seeing your slides.
Yeah, I need to get back to it. Sorry, I forgot to click on the share. There. So we can distinguish in biology 2 meanings of function. The selected effect definition of function, which is what the structure was selected to do. So the heart was selected by natural selection to pump blood. Organisms which had a heart which was better at pumping blood were kept by natural selection. Organisms whose heart was less good at pumping blood were not kept by natural selection. There was no selection to make the heart easier to listen to by doctors. There was no selection to allow the heart to have more heart attacks. But these other roles exist. And so that's what we call the causal role definition of function, which is function is what it does, whether it was selected or not. And for genes, this is very important for genes and genomes, because, for example, if all this discussion started with the ENCODE project, which looked at RNA-seq and ChIP-seq all across the human genome, and if you find somewhere where there's transcription or somewhere where a transcription factor binds, this is in a way a function, but it doesn't tell you this is the function it was selected for. It doesn't tell you this is what it has to do for the organism to survive. But yet it is, we study it and it is somehow relevant. And if you look at medical genetics or even the function of genes, say in fly, where we know from mutants, a lot of the functions we know are the function you get when it doesn't work. So we have these 2 roles, the selected effect definition of function and the causal role definition of function. And the only one which is interesting for evolution, for comparative genomics, is the selected effect. Now, a point I want to speak about first when I speak about function information is biocuration. What is biocuration? It is verifying the information we put in databases. So all of you have used databases, but it's not magic what's in it. It had to be put here, and it can be put in several ways. And one way is what we call uncurated databases. So an uncurated database, for example, NCBI nucleotides, which we also call gene bank for historical reasons. There's all the nucleotide sequences which were ever sequenced and published are there, all of them. So if something is wrong, it's there. If something is redundant with something else, so several people sequence the same gene, it's all there. There's no organization. If someone put the wrong species when they entered it, it's the wrong species. If someone wrote the function, instead of writing it in the function field, wrote it in another place, it's there. It's very lowly organized, and you cannot trust the knowledge there. On the other hand, it's complete and up to date. Every night, GeneBank is updated. You know that all the new sequences are there, and everything which was ever done is there. The other type of database, a curated database, of which the most famous example is SwissProt UniProt, so the SwissProt part of UniProt. And their information is verified by humans, who we call biocurators. So people read the information, verify it, and put it into the database and approve it. So things are verified, are trustworthy, there's very low redundancy, annotations are standardized, so it's much easier to compare things because everyone uses the same words because professionals putting this in, and it's organized and reliable. On the other hand, it is not necessarily up to date because humans take time to do things. So biocuration definition is the translation and integration of information relevant to biology into a database or resource that enables integration of the scientific literature as well as large data sets. So when we say the scientific literature, for example, at SwissProt, the curators read on average 1 paper per day per curator to add this information that then you can access automatically. I've been saying annotation. What is annotation? It is associated a biological object, for example, a gene, to a feature, for example, a function. So for example, if I do a blast of a gene of unknown function to SwissProt and I find a gene ontology term function, I can associate that term to my gene of unknown function through the blast hit. That would be an annotation. I'm carrying the function from one gene to the other, but it's uncurated. It's directly, automatically from the blast hit. I can also read an article which made experiments on this gene and say, aha, it has this function from the experiments. Then that would be a curated annotation. In both cases, annotation, which is associating a feature, for example, a function, to a biological object. In our case, what interests us is genes. Now for annotation, you can use anything, and historically the start of SwissProt, which was the first annotated database, was with free text, which means people write and people read, but it has limitations when you want to do this large scale and use it automatically, like we would want to do in genomics. So to solve this, we use what is called ontologies. So what is an ontology in bioinformatics? Ontology is first a list of terms, so we agree on the terms that we use. If we have just a list of terms and nothing else that we agreed on, it is not an ontology, it's a controlled vocabulary. Then we have definitions of the terms. So maybe we have the term enzymatic activity, then we have a definition. What is enzymatic activity? It's the activity of a protein which catalyzes some chemical reaction. And if I have a list of terms with their definitions, I have a dictionary. And what makes it an ontology is that we have relations between the terms. So I'm going to say that hydrolase activity is a type of enzyme activity. And that allows us to do reasoning. That is to say, if I want all enzymes, I don't only take those which have the annotation enzyme, I also take those which have the annotation hydrolase. This is a very simple reasoning. We can do more complex reasoning. But what's important is it allows us to organize the huge knowledge we have in a way which allows us to manipulate it automatically. So the most famous ontology in biology is the gene ontology. So here's an example from the gene ontology. You see that cell migration in the hindbrain is a part of hindbrain development and is a cell migration. So we have different types of relations. So I hope you see my mouse. Sorry. So here you have in blue part of, and black ISA. You have other different relations which are not used here. So cell migration is part of hindbrain development, which is part of brain development, which is part of head development, but also part of central nervous system. So here we see that it's a graph, not just a tree, because you can have several parents. And you can also have several offspring. So one term can have several relations, both up and down if you want. That's because biology is complicated. So we want to represent all this information. And then this can be used either directly manipulating the graph or just taking out the terms which we're interested in. For example, if you go to UniProt, for each gene, you will see on the function all the gene ontology terms. You notice here that it is separated in molecular function and biological process. I'll come to that in the next slide. And you see here the source. So how was it annotated? Here it was annotated by the curated database ZFin, which is a database for zebrafish. Here it was annotated by similarity, which means just has a similar sequence to another gene, and so on. And the gene ontology has actually 3 ontologies in it. Molecular function, which you can imagine as what the gene does when it's alone in a test tube. Biological process, which is what it does in the organism. And cellular component is where is it in the cell. Relative to what I was saying at the beginning, the gene ontology does not describe all the possible ways you could want to describe function. It describes selected effect functions. So what we think the gene or the protein was selected to do. So for example, the gene ontology does not cover pathological rules. You don't have a gene ontology function making lymphomes. It does not either cover experimental conditions, evolutionary relations, importantly to us, so this we must find elsewhere, or gene product type. For example, if you have an RNA which catalyzes a reaction or a protein which catalyzes a reaction, this is the same function for the gene ontology. It does not distinguish them. There are many ways we can use the gene ontology. I just showed you, for example, they are used in annotations in UniProt to make things consistent and comparable between databases. So UniProt took some annotation from the zebrafish database ZFin and put it into UniProt. And since both databases use the gene ontology, it can be easily coordinated. But one of the big uses we have for it is what we call gene list enrichment. So in genomics, we often get gene lists. You look for all the genes which are differentially expressed between 2 conditions. You look at all the genes which don't have an ortholog in another species. You look at all the genes which were duplicated. You look at all the genes which were clustered on a chromosome. Whatever you do in genomics, you end up with usually not 1 gene, not 2, but many genes. Now, if I have many genes, it's hard to read all the papers on every gene and come to a conclusion what they do. The gene ontology or other ontologies give us tools to allow us to get to an automated understanding of what gene lists do. Now, I'm just going to take a short break here because I hear lots of alerts and I want to see if they have to do with messages for me. No, so okay, no special messages for me. There. So how does gene list enrichment work? You have a biologically relevant terms, so annotations to your genes. For example, gene ontology terms. So I have genes and they're associated to something I'm interested in. I have a list of genes which to me are interesting. Maybe it's the genes differentially expressed between young and old. Maybe it's the genes which are differentially expressed between male and female. Maybe it's the genes which are found in all mammals except human, whatever. And I have associations between my genes and my terms, which are the annotations. And now my question is, in my list of interesting genes, are these associations for some terms more frequent or less frequent than expected by chance? Are my genes which are, for example, found in great apes but not in other mammals more often involved in behavior? Are genes which are lost in birds more often involved in walking? So I'm going to try to find these associations. These are stupid examples, but I have better ones. So the principle is very simple. So this I stole from the head of the Gene Ontology Consortium. You have a reference gene list, which is the genes which are not only your genes of interest, but all those your gene of interest come from. So for example, if I'm comparing human genes which have an ortholog in chimpanzee and human genes which do not have an ortholog in chimpanzee, my reference is all the human genes. But if I'm comparing among human chimpanzee orthologs, those which were selected in humans, those with not under selection in humans, my reference is all the genes for which I have a human chimpanzee ortholog. And among these, some are annotated with your gene ontology term of interest, and some are not. For example, here, my gene ontology term of interest is the dark blue. And so I have the reference, which is all the genes I was able to study, and my gene list of interest, which is the ones I'm really interested in. For example, the ones with selection in human. And so I ask, is there more dark blue here than expected if I took this number of dots by chance here? Oops. And for this, we simply do a contingency table where we have whether the genes have this annotation or not, whether they're in my set of interest or not. And this will sum up to my background, my reference set. And from this contingency table, I can do a Fisher's exact test, a chi-square test, a binomial, hypergeometric. Usually we do a Fisher exact test, any test which tests for non-independence. If there is independence, it means that my set of genes of interest is random relative to this gene ontology term. If there's non-independence, it means it was not independent. I get a very low p-value, and I can get a ratio of how much I get relative to how much I expected. And that's an enrichment. And for example, you can do this on the Panther database website, which is the one recommended by the Gene Ontology Consortium and the one we'll use for those of you who do the exercise this afternoon. And so you get something like this, where you have the gene ontology terms of interest. As there are relations between them, you see that the hierarchy is kept, that metabolic process is a type of organic substance metabolic process, and so on. Oh, sorry, it's the other way. Organic substance metabolic process, a type of metabolic process, lipid metabolic process, a type of organic metabolic process. And for each, you can get number of genes which were in your list, in your background, how many you expect in your, how many you observed in your foreground, how many you expected, how many more you had. So you see here, by chance, I would have 1.4. In fact, I had 17. It's a 12-fold enrichment. That's an enrichment, so the sign is plus here, not minus. And it's very significant, so the p-value is very small, 10 minus 9. So now let's, after this short introduction about annotation and gene ontology, go to another question on WooClap. Do you trust information from databases? So you can reply honestly, although I was speaking about curation, huh? So either this is a bias of young people, or it's a bias because of what I was telling you, because I know that quite a few more senior researchers do not trust annotation databases. I've had people who tell me, no, the gene ontology is all crap. But you seem pretty confident. That's good. Oh, it's going down. It's going up. I'm influencing you. It's terrible. I should not speak. Huh? New poll. Do professors know to stop speaking? No. Okay, I think we reached an equilibrium for now. Going back to my slides. What time is it? Yeah. I hear something in the microphone.
Yeah, Marc, there's a question which website you were referring to earlier.
The Panther database website. I can put a link in the, if someone puts the question in the live doc, I'll put an answer at the break.
Okay.
Thank you. Okay, so now let's speak about the database that we make in my lab, which is curated. So I'm very happy that the majority of you trust curated databases. And Bgee is a database of gene expression, and our aim is to help people, biologists, understand and use gene expression so that we get an expertise on gene expression and share this so that gene expression is easier to use for you, because gene expression is a very complex object. It can be seen in terms of amount. Genes are more or less expressed. In terms of presence, absence, genes are expressed or not. In terms of relative expression, genes are overexpressed in some conditions relative to others. Gene expression can be seen at the level of an organ, a whole body, a developmental stage, a sex, an age, and so on and so forth. So we try to organize this. And so the first thing we do in Bgee is that we curate, we verify manually only wild type healthy gene expression. This means that we look at all the available gene expression data sets, and anything which comes from a tumor, a knockout, a treatment by some chemical product in the water of the frog tank, and so on, we do not use. That is because what we want to capture with our gene expression is the selected effect function of the gene. So we want to capture what the gene has done historically in evolution. And so for this, we want to be as close as possible to the evolutionary conditions and not, for example, a knockout mouse or a tumor, which is not the selected function of the gene. So wild type healthy gives us information on the selected function of the genes. It's evolutionarily relevant. We can compare between species. If I compare the gene expression in a human lung tumor and in the lung of a mouse which had a knockout of a gene, I don't know what I'm comparing. I'm comparing some weird stuff. If I compare a healthy human lung to a healthy mouse lung, then this makes much more sense. And it allows us, actually, we built it for evolution, but we find now that it's also useful for biomedical studies, because when you study, for example, cancers, you want to know how is it when I don't have the cancer. And so we serve as a reference for biomedical studies to know what is indeed the healthy expression. We annotate gene expression. We annotate it to 5 features. I only put 4 here because, no, to 4 features, sorry, yes, indeed. So one is, because the 5th is the species, but I didn't put it here. So the most complicated one is anatomy. So I'll come back to this, but we use an ontology which describes in detail the anatomy of every organ down to the cell level, because gene expression doesn't mean anything to say this gene is expressed in zebrafish. You have to say where is it expressed. Secondly, we have an ontology separate per species of development and life stages, which goes from the egg to the aging old adult. And so allows us to say this gene is expressed in the liver of the juvenile, in the liver of 45-year-old, and so on and so forth. Sex, which is pretty straightforward in most species, males, females, although some species like C. elegans are a bit more complicated. And when we have the information, strains, that is, for example, black cyst in mouse, or for humans, different population origins, when we have this information and when we're allowed to share it, because sometimes for humans, there are confidential parts of the data. For the anatomy, as you can see on this slide, it is rather complicated. Anatomy is a complicated thing because you have the different structures, which there are many, and which have different terminologies. So we have to put together the terminology, for example, for humans from medicine and for various animals from zoology, which might use different terms for the same thing. And the relations are very complicated because things can be inside other things, but they can also have the same function as other things, or they can be developing from other things. So for example, the cerebellum is physically in the head and is functionally part of the nervous system. And then we use this data that we curated to be wild type healthy, these ontologies we have to annotate to integrate all the different types of data that we have. Because for gene expression, you have many different ways to measure it. And we want to integrate all this together between different experiments, between different data types, between different species, so that you can use it in a transparent way all together. So we took historically ESTs, which are the first type of measure of gene expression, then microarrays, and we continue because there's still microarrays available, surprisingly. More and more, of course, RNA-seq. I mean, now mostly RNA-seq. Also in situ hybridization data, which continues to grow, and there are whole projects of systematically doing in situ hybridization in, for example, the embryos of mouse or zebrafish. We integrate all this data with common standards of healthy wild type, quality control per data type, but always with consistent criteria. We reanalyze everything to say when a gene is expressed or not. We map all this to the same ontologies, notably Uberon for the anatomy, which is the most complicated one. And this is what we integrate into Bgee. So all the different data types are integrated together so that when we say this gene is expressed, we took all the information available. And how do we integrate? Because these are very different data types. So the first level of integration is that for every data type, we call whether a gene is expressed or not expressed in a given condition. So a condition will be an anatomical structure at an age or developmental stage, in a species, in a sex, in a strain. course, we don't always have this information. So sometimes we might know it's adult mouse liver, but we don't know the exact age nor the strain. So that's why ontologies are useful. You can map to more or less precise levels. In any case, we always call present or absent. So for in situ hybridization, that's the only information you have. All the others are quantitative, but you can say, you can define a background noise level, for example, for a microarray, and say everything above this background noise level is expressed. For RNA-seq, we also define a background noise level by mapping reads to non-genic regions, intergenic regions. And this allows us to say what is expressed more than the background noise is present. And then we can integrate all this. If we have different data types which all say this gene is expressed in the liver at different ages, we can then put this together and say this gene is expressed in the liver at all these different ages. We can also try to integrate a more quantitative aspect, because it's also important to know if a gene is more expressed or less expressed. And for this, for every data type, we do a non-parametric ranking. So we can say this gene is more expressed than the others or less expressed than the others in a given condition. And that you can do for every condition, every anatomical structure, every age, and so on. But then if we want to take the gene's point of view, we can say for this gene, in which anatomical structure, in which age, in which sex, did it have the highest rank? If a gene has the rank 10,000 out of 20,000 in the liver, but in other organs it has rank 15,000 out of 20,000, I'm going to say it's all the same, more important in the liver than elsewhere. And so then we integrate these rank scores across all the data types and all the conditions to get a general idea of where a gene is most expressed, and in that way to get an idea of where this gene is most important. We integrate the scores by weighing by the amount of information, so that you know that RNA-seq, which brings more information, will actually bring more weight in the end than, for example, in situ hybridization, which has very low information on this score. So for RNA-seq, I'll just do one short slide to explain how we do, because this is the less classical one. Most people use RNA-seq simply to do quantitative comparison, but not to call present absent, or if they call present absent, they put an arbitrary threshold of, say, TPM2 with no justification. So our starting point is that it has been found that when you look at RNA-seq distribution over genes, what you get typically is a kind of bimodal distribution with a smaller shoulder here and a big peak here. And this big peak is in fact the genes which are actively expressed. So it's not just that they are expressed as in the polymerase was around there and made some RNA, but actually that there is active opening of the chromatin, transcription factors binding, and a function for the gene. And here, this shoulder is more the background transcription of the genome. And what you can see is that if you look at intergenic mapping of reads, it overlaps a lot with this background of genic expression. So we define for every species a reference intergenic. So this is from an old study. We have refined this, but my graphs are more complicated, so I put the old study here. We refined the intergenic for every species to define a real intergenic, which we guarantee is not genes. For this, we analyze all available RNA-seq. This is important because in non-model species, genes and intergenic are poorly defined. In human or mouse or Drosophila melanogaster, this is not a problem. You go in even another Drosophila or another primate, and suddenly lots of genes are not annotated, and what we call intergenic might include a lot of non-annotated genes. So we define a reference intergenic, and then for every library, you map the reads to the reference intergenic, to the genes, compare the two, and those genes are more expressed than the reference intergenic, we call expressed. We actually have an R package, I will put you a link later, which allows you to do this on your own data sets if you want. And so in the end, when we integrate all this information, you get a page of a gene like this. So this is the gene APOC1. It's a liver-specific gene in human. And here you have, first you have all the anatomical structures where this gene is found present. And you see there are 217. So this means it's a very widely expressed gene. So a gene which is found in many structures. But yet we know from its function that its most important function is in the liver. And indeed, if we look now at the scores we have from integrating the levels of expression in all the different organs, we see that the top score is in liver, and the next is right lobe liver, and the next is a gland cortex. So we have here this highest expression in the liver across 14 developmental or aging stages. So we can both find all the structures in which this gene is expressed and find which is the top one. And we can do this in every species for which we have data, so that you see that in different species, the top expression information for this gene, for the ortholog of this gene, is always liver. In mouse, in zebrafish, in chimpanzee, in macaque, in rat, in dog, in guinea pig here. I stopped because of the limit of the screen. You see that always we have one top expression in the liver. And it's from various information sources. For example, here it's Affymetrix, and here it's Affymetrix and RNA-seq. Here it's Affymetrix in situ hybridization and RNA-seq. For the non-model species, sorry, it's usually only RNA-seq because the only thing we get from non-model species. So let's go back to do a poll. You can already follow the link. I'm going to share the screen soon. So what would you compare expression in human lungs to? Answers are less homogeneous than in previous. This is interesting. Tuk. What will win? Nobody, no love for the gills. You know that gills are what allow fish to breathe, huh? No fisher, no people who do fishing in their spare time in this group. Okay, it's stabilizing. Oh, someone was influenced by what I said. This is interesting. So I don't know how many of you have studied zoology, but fish don't have lungs. That's an interesting observation. A zebrafish does not have lungs. So if you're going to want to compare to the zebrafish lungs, you're going to have a tough time. That's where it's interesting that experts are annotating databases and sharing their expertise with you, because of course you cannot know everything, right? So it's useful that we provide you information so that you can find this information even when you, of course, did not study everything in biology, or if you study it, you don't remember it. So when we compare expression, we want to compare homologous anatomical structures. This is exactly the same logic as comparing orthologous genes, right? You want to compare what is comparable. So when you compare the expression between two species, you want to compare the orthologous genes, but you want to also compare their expression in the homologous anatomical structures. So if you want to compare, I actually don't have the lung here. So if you want to compare the human lung to something in a fish, the homologous structure is actually the swim bladder. The swim bladder in teleost fishes is an organ which allows them to change the buoyancy, so how dense they are relative to the water. So they put more or less gas in it, and it allows them to float up and down in the water. And this actually derived in teleost fishes from an ancestral organ which allowed to exchange inefficiently gas with the atmosphere. And in the ancestor of tetrapods, such as us, land vertebrates, this inefficient organ was selected to become more efficient and eventually became the lung. And in lungfish, which are actually closely related to tetrapods and not to teleost fishes, you have both. You have the gills, which allow to exchange oxygen with the water, and lung, not so efficient, but allows them to survive when there's no water. And so if you compare the human lung to something in zebrafish, it should be the swim bladder if you want homology. If you wanted the same function, it should be the gills. If you look for the lungs in zebrafish, you won't have much luck, there are none. And so one way to do this is that we have put into the Uberon ontology of anatomy or the knowledge we have from the literature of homology at the anatomical level. And we did not do this by comparing the expression of everything against everything, because then it would be circular. If we use the expression to know what should we compare, and then we compare after that, then we are comparing what by definition had the highest expression similarity, and we don't know what is the biology here. We're just knowing that what's the highest correlated is the highest correlated, it's circular. So we want to have external information. So we read the literature of paleontology, of developmental biology, of comparative zoology to know which anatomical structures are homologous between species all the way down to the level of animals, metazoa. And at every taxonomical level, like the taxonomical level you can have in the hierarchical orthologous groups, we have a level of homology at the anatomical level. Now, this makes a horrible file that you don't want to read, but we made a tool on the web so that you can do this easily. So if you put orthologous genes here, you will have, so here are different genes which are all known to be brain specific from the literature. I mean, we know the human brain is human gene is brain specific. We put the orthologs in various mammals here. We search and we get that that all these 13 orthologs are all expressed in the brain, and that's also where there's the biggest score among these genes. And the next is the central nervous system. So there's conservation of expression of this gene in the brain of various mammals. And here we can guarantee you that we took the homologous structure. So the brain for mammals is kind of trivial, but if, for example, you started with human data and you wanted to find the comparison in zebrafish because you are interested in verifying some biomedical hypothesis in zebrafish, it would automatically take you only the structures which have homologs in zebrafish. I spoke before about the gene set enrichment on gene ontology, but I said this is in principle something you could do with anything where you have an annotation. So we can also do this with our annotation of genes expressed in anatomical structures. And that's what we've done. We've created a tool called TopAnat, which allows you to find the top anatomical structures for a gene list and whether they are significant. So this principle is exactly the same as a gene ontology test, but instead of associating a gene to a gene ontology term by annotations such as in SwissProt or in the model organism databases such as MGI, ZFIN, Flybase, and so on, we associate a gene to an anatomical structure by gene expression. And so for each anatomical structure, for example, for brain, you would have in your gene list of interest, for example, genes selected in human, those are expressed in the brain or not expressed in the brain. And for the genes, the other genes, for example, all the genes for which you could find orthologs on which to compute selection, you will also have whether they're expressed or not expressed, and you can do a Fisher test, a hypergeometric test, a chi-square, and so on. As usual, we have implemented only Fisher and hypergeometric. And both for the gene ontology and for Uberon, as I told you, we have relations between the terms. So it means that the comparisons are not independent. So we actually, I'm not here putting slides on the detail of how this works mathematically, but we allow what we call a deconvolution, which allows you to make these comparisons more independent. And so on the website, it looks like this. Here are genes which were chosen that when you knock them out, you get a phenotype in the fin, in the pectoral fin, in the zebrafish. So you knock out this gene and the fin looks weird. You put all these genes into Bgee TopAnat, and we run this comparison, and we find that they are 7 times more frequently expressed in the pectoral fin than expected by chance, and this is highly significant. Very low p-value, very low false discovery rate. And so you can do this on any gene list you like, and on all the species for which there is data in Bgee. A disadvantage relative to the gene ontology is that you don't have all the aspects of function, you only have expression. An advantage is that if you are working in a species other than, say, mouse or Drosophila melanogaster, which are very studied, we guarantee you that this is the gene expression expression function in that species. We only use the gene expression curated and annotated in the species you are studying. Whereas with gene ontology, because most annotation is done by orthology, if you look at the function of genes in, say, chimpanzee, you're actually looking at what was the function of these genes in mouse or human, because that's what was studied. No one studied function in chimpanzee. If you're looking in flatfish, no one studied in flatfish. You're having the zebrafish or mouse data, or maybe in fly. For the expression, if you do here the expression in chimpanzee, it will give you only the expression in chimpanzee, even if there's specific different expression in chimpanzee than in other species. Both TOPANAT and gene ontology enrichment analysis have pitfalls you should be careful of. And the number one, which is really important and I cannot insist enough, is the background. If, for example, I look at genes which have orthologs within human and chimpanzee and have selection in human, my background should absolutely be the genes which have orthologs between human and chimpanzee. If my background is all the human genes, then I am testing not genes which have selection relative to genes which don't have selection. I'm testing genes which have an ortholog and selection relative to genes which either don't have an ortholog or don't have selection. And if a lot of genes don't have an ortholog, that's the main thing I'm testing. So you have to be super careful about this. It might be the genes for which you can find an ortholog. It might be the genes which for your microarray. It might be the genes for which mapping is possible in your RNA-seq experiment, but you should always be very careful about this. You test many gene ontology terms on many anatomical structures, so there's multiple testing. We provide you an FDR measure, so there's the PantoDB tool, which I'll give you a link afterwards. And there are non-independence of terms, which means that, of course, cerebellum is in the brain. So if there's more expression in the cerebellum, there's more expression in the brain. Of course, hydrolase is an enzyme. So if there's more hydrolase genes, there's more enzymes. So for this, there are algorithms to decorrelate this non-independence, which come from the package TopGo, which can be used, as its name indicates, on gene ontology, and which we use in TopAnat on anatomy. The Bgee data is available from the website, what I've been showing you. And we also have R packages, which I'm not demonstrating today because that would be a whole different course. But we can put you a link to a course we gave on specifically only Bgee in June. If you want to use Bgee data, go wild. It's CC0, which means there is no copyright at all. You don't even have to cite us legally. You can just take it, repackage it, and sell it under your name, and it's legal. It's just not very polite. So we ask you to please cite us because that's the polite thing to do among researchers. And there are packages with the links here, which you have in the PDF I gave you. For the first package allows you to recover Bgee data and do TOPANAT. And this one allows you to call present absent from RNA-seq according to our methods. And so that was my presentation of Bgee. I will now look at the questions, and we can also take a short coffee break if you want, but I will first look at the questions. So I will stop sharing. Yeah. So let me see how do I see the questions now? Okay. So there are questions in the Slack, in the Zoom, sorry. Good morning, good morning, good morning. Could I provide the citation? I think it's on the slide. Let me check because I gave you the PDF slides, right? So the citation should be on the slide. Let me check. Yes, so it's on the slide. So Rob Waterhouse says that the point I make on background is very important. Thank you. So I will put, okay, Rob put PantoDB here, but I'll also put in the live doc. Okay, a question from James Ord. Many gene expression experiments will have data from multiple treatments. Are all the data incorporated into the annotation or just the data from control conditions? So for Bgee, only the data from control conditions. So if on the one hand, if you took some, I don't know what animal is interesting to you, say you took some flies and some of them you put an endocrine disruptor on them, or you put them in a very high heat or something, and others you left them alone as a control, we will only take the gene expression from the control. So this allows us to cover as many gene expression data sets as possible, because many data sets where there's an intervention, there's also control, hopefully. but to guarantee that what we integrate is only the ones with wild type healthy, non-manipulated expression. Ta-da. What's the difference between intergenic and intron? So introns are part of genes. They are transcribed. So the definition of an intron is that it is transcribed and then spliced out. So there is as much RNA coming from introns as from exons produced in the cell. It's just used differently. Intergenic means it's not in a gene at all. So it should not be transcribed if there was 0 errors in the machinery. If the cell functioned perfectly, intergenic regions would never be transcribed, right? So if you define a gene, definition of a gene is a whole other question apart from definition of function, but say let's, for our purposes here, define a gene as something which is transcribed to make either a functional RNA, like a ribosomal RNA, a microRNA, and so on, or to make a protein, then intergenic is what is not in any of these genes. And within these genes, within the boundary of these genes, there is not only, for example, for protein coding gene, everyone knows protein coding genes. Within the boundary of the gene, there is not only what codes for the protein, there is untranslated 5', which is transcribed but not translated, then the translation starts. You have exons, which include the untranslated and the translated part. Then you have introns. So you will have transcription of all this, then splicing out of the introns, and then translation will start at the translation start site and go to the stop codon, leaving at the 2 ends the 5' and 3' untranslated regions. All this, it is legitimate to get RNA from. All this was transcribed, which is what makes RNA. Whereas the intergenic regions are regions which are not transcribed at all. If I found transcripts mapping into intergenic region, does it mean the gene is misannotated? No, because the transcription machinery is not perfect. There is noise in the cell. Processes are noisy. So what we do to define reference intergenic is that we take all the RNA-seq libraries available in healthy conditions for an organism, all of them, across all conditions, all sexes, all anatomy, all ages, and we map them all. And then we, if there are regions which are annotated as intergenic in the genome, but we find actually a lot of reads mapped there, and we do, especially in non-model organism, then we say this is suspicious. We don't think it's a true intergenic. We think there was a misannotation, and in fact, there should be a gene here. Now, it's not our job to improve gene annotation, so we don't do that for now, but we just put it aside. And now those regions where there is no gene annotated and there are very few reads which map over all libraries, all experiments, those we call reference intergenic. And that's what we use then for each library to estimate the noise. And also there is what Rob said, that it could be low complexity reads. That depends on the parameters of your mapping, obviously. How often do you update the database with newly submitted data? So now I will go away and not, so no, the serious answer is not often enough. No, we work continuously on updating behind the scenes, but we push updates to the web every 1 or 2 years, and we are trying to do this faster now. So we have one which came out earlier this year and one which come out at the end of this year, and another probably in the spring 2021. I should also say we make small updates where we add data to the species which are there, and big updates where we add new species. So right now we have 29 species, and the next big update will have more than 60. What would be an estimate of outliers of data set of a given tissue? Would you provide a flag for these data sets for other researchers? Stefan Butz, could you take the mic and explain your question more, please?
Yes, sure. Just on maybe if you're analyzing, I don't know, over time 20 testis data sets for mouse gene expression, would you see often outliers occurring where you say, okay, this is for sure not a testis, or this is maybe bad quality, and would you then maybe have like a kind of a blacklist of data sets where you say maybe don't trust this data, maybe go to these?
Yes, so first for the end of the question, how we get there, we do have a blacklist, and it was on project to publish it separately. We haven't yet done this because it's kind of delicate to publish an official list of don't trust these data sets. We're discussing also with the people in some other database, like Swissprot also has this experience that there are papers they know are false. Should they say so? How we do this is data type dependent. So we have different quality controls. Such clustering as you described, we do in single cell, which is not yet available on the website right now. It will be available early in 2021. For microarrays, for example, we have a quality control which correlates very well with your question, but is not how we do it. We do it rather by a measure of the dispersion of the genes. I'm not going to get technical here. The thing is, the danger with what you're saying is that we have different conditions, and some we capture in the annotation, and some we are not able to capture. But biology is very diverse. So suppose we would have data from many virgin fly females, and now we get data from one mated fly female. And the expression is different. And we didn't capture the fact virgin or mated. Then we should not say it's wrong. It's also a bona fide female fly. It's in a different condition. So we see this when we dig, that there are things which look like outliers, but there are different conditions. So we use other criteria which often correlate strongly with the fact of being an outlier, but are more objective than that. Otherwise, it's very difficult. Do we have some database in mind to reckon out plants? This is like my most frequent question, and no. I'm sorry, I don't have enough funding to do it also for plants, and I don't know anyone else doing it. But yeah, it would be cool. Could you quickly go over the importance of the background and why is that important? Okay, so I will share a slide again. Wait a second. Or wait, I will try something. I will try to use the whiteboard here. Let's see how this works, because I hate drawing on these things, but let's try. Okay, you all see my whiteboard? So now, what we do, we have a contingency table. You see that I draw super well, where we have what interests me and what doesn't interest me. And I have a function or an anatomical structure F and not having this function, right? And the sum of these two is what I will call my universe, which is everything, or the background. I draw super well, it's a pleasure. I feel like I'm back in kindergarten. There, okay? Now I'm doing a contingency table on, let me change color. How can I change color? I don't know, okay, too bad. I'm doing a contingency table on this. This is from my measures, that's cool. This here is the difference between these two sides. Everything I have and what is in my list. So what's in my contingency table depends strongly on what I call my universe. So let's suppose that this is all the genes in the genome, and these are the genes which have a certain function. And now I'm looking at orthologs between two species, say between a human and a bird. And in bird, there's a big bias because there's GC-rich chromosomes which are very hard to sequence. So there's parts of the bird genomes for which I don't manage to get ortholog. And maybe this is biased. Maybe these genes have less my function than other genes. So it's like this. Okay? So now, if I compare this frequency to this frequency, I have one result. But if I compare this frequency to this frequency, I have another result. Okay? And so, for example, to give an example we had for real, the first time we tried to look at, we looked at terms which have a gene ontology related to testis function. And we found many of them expressed in ovaries. That was weird. And then we found out that that's because there is a lot of gene ontology annotation of breast and ovary cancer data sets, which means that there's a lot of function annotation for female reproductive system and much less for male. And so the genes which are in the gene ontology, if you take just all genes in the gene ontology, not looking at what term, just does it have a gene ontology annotation of some detail, and look where they're expressed, they're more expressed in ovary. So our background was wrong. Our background should not be all genes, it should be all genes which have a gene ontology annotation. And when we did this, we recovered that they were more expressed in testes, but not in ovary. These genes which had a function in testes, which is what we expected. I hope this makes sense. A flag for data sets which reflect the tissue the most. This is an interesting question. I'll think about it. It's always delicate to tell people which data sets are good or bad, because the data sets are made by people, are published, are part of students' CV, people's careers. It's an interesting question. I'll think about it. What I can tell you is that if you're interested in human, the biggest data set in human is GTEC. And the GTEC data set is very good, but is not directly curated by GTEC. They take everything that the pathologists give them. So if they say, give me this piece of brain, and the pathologist says, here it is, they use it. But the pathologists also write a report. So the curators of Bgee read all these reports, which was a lot. And we removed about 50% of the GTEC data, which is not from healthy individuals or not actually what they say it is, because the pathologist wrote, I tried to take the structure, it was hard to dissect, and I also got a bit of the neighboring structure. But you also have things like the brain of someone who has Alzheimer, the liver of someone who has a liver disease. We removed the whole samples entire of people who died from drug overdose because we said the gene expression is not normal. So you can access through the Bgee R package, for example, the subset of GTEC which we curated as healthy and well annotated. That kind of half answers your question, I think. Okay, so 5 minutes later, I'm ready to restart. I hope you are. I'm just setting up the right things. So part 2, we're going to more comparative genomics and evolution with patterns of gene and genome duplication. So let's first with a little poll. What's more common? So the race is on. Point mutation is on the lead. But duplication has a strong camp in its favor still. Seems to be stabilizing for a while. It's 3/4 point mutation, 1/4 duplication. So how frequent is duplication in evolution? Well, actually quite frequent. So this is a rather old paper, but I've checked its results have kind of stood not too bad the test of time. They took the genomes which were available in 2000. So for those of you who were born after 2000, we actually started doing genomics already. And these are the genomes we had available at the time in eukaryotes. That's all of it. That was complete comparative genomics of 2000. And for each genome, they looked for genes have a homolog within the same genome, so paralogs by definition. And they looked at their divergence. And the idea is that genes which diverged recently have lower divergence, and genes that diverged a long time ago have higher divergence. And what they found was this exponential decay here in every species they looked at. And exponential decay is what you expect if you have very frequent birth of duplicate genes and very frequent loss. And they estimated in this study that the frequency of duplication is about the same, on the same scale as the frequency of point mutation. I've tried to update this because it's from 2000. I found several papers which update. They always get to the same scale question, and then how you exactly measure point mutation frequency and how you exactly measure duplication. There's always a lot of debates, but let's say duplication is really not rare. It's on the same scale as point mutation. It's kind of surprising because I would have voted like the majority here 21 years ago. I mean, we expected duplication to be a rare event, and that's because of this exponential decay. If you don't look at the recent duplications, you see not so many, because most duplications are negative. And actually, I thought, but then I thought we don't have time in this course to have you have a discussion about the differences between what we see in evolutionary genomics, comparative genomics on the one hand, and medical genetics, medical genomics on the other hand. Because when you look at the frequency of events in diseases, you get completely different statistics, because in disease, you get the things which cause phenotypes which decrease fitness. Whereas in evolution, we see kept over the long term genetic modifications which either do not change fitness or increase it. And very rarely those which decrease it. So for example, you will never see trisomies being fixed in evolution, because if you have an unbalance of chromosomes, it's negative. But it happens, of course, in medical genetics, as we all know. Now, there are different types of duplication. I just spoke of trisomies. And here I'm illustrating them rapidly with the Hox cluster. So everyone knows the Hox cluster, I hope. It's one of the most famous gene clusters in biology. So rapidly, these are genes which are transcription factors, which are found in a cluster along the chromosome. They were first discovered in fruit fly. So you have these genes which are in a cluster along the chromosome, and they're expressed in embryonic development, notably from one end of the embryo to the other according to their order on the cluster. So that's very nice. And you find them in all animals, and in most animals it's preserved as a cluster. And I'm not going to go into the evo-devo biology of this cluster now, but just to say, how do we get this? So if we have a cluster of genes which are actually all Hox genes, so all actually homologs, it means that they are paralogs and that they came from tandem duplication, local duplication. So we had a gene which duplicated locally to another gene, and then maybe they duplicate to other chromosomes, came back together, and in the end, you got this array of genes which were duplicated in small-scale duplications, one gene at a time. But then, for example, if you take human or mouse, you have 4 Hox clusters. And these 4 Hox clusters all have, at least part of the cluster in a way that you can see that the whole cluster was originally duplicated. So here we have a large-scale duplication which duplicated the whole cluster. And actually, what we know now happened is that the whole genome was duplicated. So when you duplicate the whole genome, the whole cluster is obviously duplicated because it's part of the genome. And so now you get not 1 Hox cluster, but 2. And now if you duplicate the genome again, which happened in the ancestral vertebrates, there were 2 whole genome duplications, you get these 2 times 2, 4 Hox clusters. And in, for example, zebrafish, there are 7 because there was another genome duplication in fishes, which 4 times 2 makes 8. Then you lose some and you get 7. So these are the different types of duplication which affect most evolution. There's also like big segmental duplication, but they often lead to the kind of problems we see in medical genetics. So they're rarely kept. So what you usually get is either small scale, 1 or 2 genes, or whole genome. Whole genome duplication is of special interest to me because it allows us to study the evolution of gene duplication in a very controlled manner. All the genes duplicated in a genome duplication duplicated at the same time with all the regulatory elements. And so we can then compare, if we study their evolution, we have a controlled experiment, kind of natural experiment, where all the genes which we are comparing have the same age, the same history. And genome duplication has become a big topic in comparative genomics since the late 90s. The first eukaryotic genome which was sequenced was the yeast Saccharomyces cerevisiae. And Saccharomyces cerevisiae was sequenced because it was simple. It was studied because it's simple. It's supposed to be a simple eukaryote, and it has a small genome. And to their surprise, they found that there were lots of blocks here colored, which were homologous between chromosomes. And when I say blocks, it's whole groups of genes which keep their order. And if you paint the chromosomes with these homologous groups, you paint almost everything. This is the 97 paper, it's been improved since. But what we find is that, in fact, most regions of the genome have a double somewhere else, a paralog. So this means there was a genome duplication in the ancestor of yeast. So even a simple organism like Cerevisiae had a genome duplication in its evolution. So genome duplications happen. Of course, not as frequently as gene duplications, but they happen. And an interesting thing about genome duplication is that it duplicates everything, but then not everything is kept. I showed you before on small scale duplications, there is huge loss. There's this exponential decay. So it's not quite the same mechanism for genome duplication, but you have a lot of loss. And so what you will have here, this is from the study in 2004, which proved there was genome duplication in yeast. This suggested it in 97. This proved it by comparing to an outgroup. You've heard of outgroups. Outgroups are important. So if you had this gene order on a chromosome in the ancestor, and you have a speciation, so orthologs. In this lineage, no genome duplication. So let's say that nothing changed and you get to this. In Saccharomyces, you have a genome duplication. Now you have everything exactly double. Immediately after the duplication of the genome, all the genome is double. But now these genes which are in double, they are not always useful. You don't need both genes. So you can lose, and you lose randomly one or the other copy because they are symmetric at the start. So now you have this feature here where many genes are in only one copy. You only have one copy of gene 1, only one copy of gene 2, two copies of gene 3, only one copy of gene 4, and so on. So if you count the genes and duplicate between these two blocks, which is what allowed to first suggest a genome duplication, you have not so many. So it suggested, but didn't convince many people because it's not overwhelming. But now if I compare these two blocks to an outgroup, which is similar to the common ancestor, I find that these two chromosomes have an alternating orthology to this outgroup. On this copy 1, I have these genes which are orthologous to this outgroup, and here those genes. And so sometimes I have double, but often I only have one, but it alternates like this because these two regions of chromosome come from this duplication of genome. And thanks to such studies, we can estimate the loss and see that most genes are indeed lost after genome duplication, but quite a few are kept. Otherwise, we would never have any evidence. So the order of 10, 20%, depending on genome duplications, will be kept on the long term. So now I'm going to ask you to do something a bit different on WooClap, which is not a poll, but brainstorming. So when genome duplication was first suggested to be important to evolution in 1970, it was by Susumu Ono, who said vertebrates and especially mammals are too complicated to have developed only by point mutations. So there must have been massive duplication, which allowed the complexification of vertebrates and mammals relative to invertebrates. And so I'd like you to think about this. I'll give you a few minutes. So I'm going to go to the WooClap. Stop the previous one. Okay, so here you can write, what do you think is a higher animal? And what is the relation between genome duplication and higher animals? And you can also like the answers of other people. I'm not going to share for now. I'm letting you do it. So I see some answers on what is a higher animal. You can also answer the second question on what is the relation between genome duplication. Yeah, thank you. One answer. And as I said, you can also like other people's replies. You cannot dislike them because it's a very polite environment here. Okay, so I'll share the screen so we can look together. Okay, so some answers on higher animal, evolve more through time. 2 people have liked that, so 3 people have written it. A concept that assumes there are higher and lower organism obviating tree-like evolution. This is a complicated way of saying the same as the no such thing answer, I think. More complex genome, more complexity, subjective, more duplication, more brain, further down the evolutionary tree, intellectual. So I kind of see 3 clusters here. Those who say there's no such thing, those which say that it evolved more or longer, 4 clusters. Those which related to behavior or intelligence, and those which related directly to the genome. So more complex genome, more regulation, more redundancy. And on the relation to genome duplication, it increases variability, provides more genetic raw material, does not necessarily lead to more complex functions, provides the possibility for the same gene to evolve differently, allows for redundancy, creates neofunctionalization, allow increased expression of beneficial genes, increased possibility for evolution. So there seems to be a general trend of saying that it increases possibilities, basically. It opens doors, right? So I'll leave this open if you want, but I will go back to my slides. So whole genome duplication is found repeatedly in plants and a bit in animals, and also in other organisms. So here I have some slides on plants and animals, but for example, I showed you in yeast, there's also multiple genome duplication paramecium, which is in amoeba. So this is a review of 10 years ago, but it gives you an idea of the distribution. Here you have amoeba, paramecium. Here you have the yeast. Here you have, in animals, the only ones known at the time, 2 genome duplications in vertebrates and 1 in fishes. And here you have plants, what you see all over the place. So genome duplication is quite common in plants, actually. And if you think even about, I don't know if any of you work in plants, but if you think about plant breeding or the diversity of plants, you have many hybrids, many tetraploids or octoploids. And so some of these hybrids or tetraploids will eventually get fixed in evolution and become genome duplications. So genome duplication is quite common actually in plant evolution, much rarer in animal evolution. But, and let's say when we discovered this one at the origin of vertebrates, it's very intuitive to say, hey, we are vertebrates. We feel kind of superior to those stupid flies. And we have genome duplication. There must be something to this. So, yeah?
Sorry, we don't see your slides.
You don't see my slide. Oh, I'm sorry. Thank you for telling me. I forgot to click. I didn't click the. Oh, okay. Sorry. Back 1 slide. I said this, then this. Sorry. Okay. So start again. Here are the. So these are genome duplications in various species known in 2009. Oops. Here are the parameciums, yeast. Here are plants with many genome duplications, as you can see. And here are the animals with only vertebrates, because that's the only ones we knew. And you see 2 invertebrates here. And as I was saying, when we found genome duplication vertebrates, it was kind of intuitive to say that's linked to our complexity as vertebrates. But then that doesn't really square with the fact that there is also genome duplication in teleost fishes. There's more, which are not shown here, in other fishes. There are in parameciums, which are not known to be specially complex behaviors, for example. Plants have way more genome duplications than animals, including vertebrates. So it's not clear in any way how you could link any concept of complexity to genome duplication as far as the phenotype goes. And as far as higher animals goes, I would agree with those who said there is no such thing as a subjective thing. There is no objective definition of higher animals. Depending who you speak with, they will say it's humans, it's primates, it's mammals, it's vertebrates, whatever. The only common point people will have is to include us, basically, the person speaking. And the same would be for higher plants, something which is going to include in the end Arabidopsis, but apart from that, we're not very clear. So you should realize that all these species have evolved since the same time, right? All vertebrates, for example, have the same amount of evolution, the same time of evolution since the Cambrian to us. This is an updated slide of genome duplication in plants. You see that it's really very common. The dates are sometimes unsure, but it's really very common in plants and unrelated to any clear feature such as complexity or so there's a lot of debate whether genome duplication of plants are linked to increase in speciation rates. And to my knowledge, this is not yet solved. So if there is something, it's very weak. It's not a strong feature which would be easy to see. And for example, yesterday, Rob told you that the biggest amount of animals is arthropods and the biggest number of species in arthropods is beetles. And there are no genome duplication in beetles. So you can have diversity clearly without genome duplication. And I told you there are more genome duplications known in fishes. This is a recent paper on genome duplications in vertebrates. You see that there are 2 at the origin of vertebrates here. There's a more recent one just in one frog, Xenopus laevi. There is at the ancestor of teleost fishes. There's also in cyprinids, goldfish and carp, in salmonids, salmon, trout, and so on. And sturgeon, we're not clear how many there were. Some sturgeons are octoploid, depends on species, but it's very diverse. And sturgeons, for example, sturgeons and gars are those fish which did not have the teleost radiation. So almost all fish you can meet are teleost. This is like 99% of fish species. And these are sister groups which were not so successful. And yet this one had lots of genome duplication. So it's not very easy to relate these things. As I told you before, not all genes are kept in duplicate after duplication, after genome duplication. And this is actually very interesting because it is not a random set. So this was shown originally by a paper in 2004, which as far as I'm concerned is one of the really classic papers of comparative genomics because they showed the importance of controlling. So if you look at duplicate genes in one species, for example, S. cerevisiae, you can compare the genes which are duplicated and the genes which are not duplicated. And the genes which are duplicated differ from the genes which are not duplicated in 2 ways. One, they evolved after duplication. So anything which happens after duplication happened to them. And that's the obvious thing. That's what we're usually interested in. But 2, it means they duplicated and they were kept in duplicate. And how do we disentangle these 2 features? When I compare these genes to those genes, they have these 2 differences. And so the idea of this paper is to use outgroups. So you have the insect ortholog of a gene which did not duplicate in yeast or C. elegans and the insect ortholog of a gene which did duplicate. And now I can compare the properties of these, and these properties are independent of the evolution after duplication. So they only depend on which genes, what were the ancestral properties which made some genes be duplicated and kept in duplicate and other genes not kept in duplicate. And so one of the ways to characterize these genes is codon evolution. So I'm going to do a super rapid one slide on DNDS. So DN, also called K, depending on studies, is the non-synonymous substitution per codon. So when you change the codon, the DNA of a gene, you change the amino acid, so presumably the function which is carried by the protein. DS or KS, synonymous changes, so you change the DNA, but you do not change the protein, so presumably you do not impact the function which is carried by the protein. So in a first approximation, only the non-synonymous changes are seen by selection on the function. And so if it's neutral, if there's no selection, then the 2 rates will be the same, so the ratio will be 1. So if the protein is evolving neutrally, the ratio would be 1. If, as in most cases, there is selection to keep the function, not to break it, it will eliminate more substitutions which change the protein and eliminate less substitutions which do not change the protein. So the ratio will be less than 1. dn will be smaller than ds, or k will be smaller than ks. So when you have negative selection, this ratio is less than 1, and you can quantify. The stronger the selection is, the lower this is. So for histones, which have super strong selection, because if there's any change, you break the chromatin and you're dead, dn is essentially 0, so the ratio is 0. Okay, and some other genes are kind of important but not so important, could be 0.5, for example. And if there is positive selection, selection to change things, that there's a change in the amino acids in the protein, which is improving the fitness of the organism, improving the function of the protein, then this will be fixed by selection faster than it would be fixed by drift. And so there is a higher rate of change of the amino acids than the DNA synonymous. And so the ratio becomes larger than 1. And again, this can be quantitative. The stronger the selection to change, the higher this is. You typically see very high D/DS ratios, for example, in immune system genes which are in an arms race with pathogens. Why is this important? Because it allows us to compare the selection between different categories of genes depending on their duplication history. And so what we see here is that the genes which were kept in duplicate in yeast, their Ka in insect is lower. So they had less non-synonymous changes fixed during evolution. Those genes are kept in duplicate. And I remind you, on this graph, we're looking at the orthologs here. So there's no impact of the evolution after duplication. This is just saying the genes are kept in duplicate are the genes which were under stronger purifying selection. The K is lower. The same in C. elegans. The same is also true for the fact of getting insertions and deletions. So the genes which are kept in duplicate are not random genes. They are genes which are under stronger purifying selection. And there's also differences in the function and in the Ka between the genes which were K or Dn, the non-synonymous rate, the genes were kept in duplicate in small-scale duplications or whole genome duplications. Okay, so the whole genome duplicates have even lower Ks who are under stronger purifying selection. So the genes did not duplicate were under weaker purifying selection. Those which were kept under duplication after small-scale duplication were under a bit stronger, and those which were kept after the genome duplication were even stronger. So you see that if I look at the evolution of the function of genes after genome duplication, I should take into account this is a biased set, also biased in terms of function. So this is a very naive with 2004 gene ontology test. And we see that there are functions which are much more common. For example, here in the whole genome duplicates, there's much more structural molecules. And we see this also, for example, in fishes. So this is a study we did as a follow-up to this. And we find notably that there is much more developmental genes and behavior genes, neurological genes kept after genome duplication in fishes, which kind of goes in the direction some of you are writing about, saying that the genome duplication allows more complexity of behavior. At the same time, as humans did not have that genome duplication, so we should temper this. And there's also the same observation that the genes after genome duplicate, which were kept duplicated in fishes, they're orthologs in other species which did not have this genome duplication, here human and mouse, have lower non-synonymous substitution. Okay, so here it's higher bar for the lower Ka or Dn. This duplication bias was studied in the early 2000s. I just showed you on evolutionary rate of genes, but it's also true of other features. And notably, we looked into gene expression more recently. And so this is, in fact, that's how we invented TOPANAT, to make the study. So sometimes the biology motivates the bioinformatics, sometimes the bioinformatics allows the biology, the to and fro. And so we looked at where genes are expressed, which are kept after genome duplication in fishes. And we find that these are all the anatomical structures which are enriched. And if you look, those in bold are all in the nervous system. And there's a huge significant enrichment of genes which are expressed in the nervous system kept after genome duplication in fishes. And this is also seen if we look quantitatively at the level of expression. The genes which are kept in duplicate here are much more expressed in the nervous system than the genes which are not kept in duplicate. And this is also true for the orthologs in mouse. So it's not because they evolved after duplication to be expressed in nervous system, it's because those which were preserved after duplication were already expressed in the nervous system. Otherwise, we would not see that expression in mouse. Now, this asks an interesting question of causality, because we know from the old studies that the genes kept in duplicate after genome duplication evolve slower. And there's also studies from the early 2000s which show that genes which are expressed in the brain in vertebrates and also other animals evolve slower, because there is selection against misfolding of proteins. So selection, very strong selection on proteins expressed highly in the brain, because the cells in the brain do not divide during life like in other organs. And so it is very difficult for these cells to get rid of misfolded proteins. So there's very strong selection on these proteins. So we have genes which are kept after duplication, which have low evolutionary rate, genes which are expressed in the brain, which have low evolutionary rate, and genes which are kept after duplication, which are expressed in the brain. Which way does this go? So what we expected is that either the genes which are kept after duplicate always have low DNDS and thus are enriched in genes expressed in the brain, or that the genes which are kept after duplication are always expressed in the brain and thus have low DNDS. What we find is actually a weird mix, where we have the genes which are expressed in the nervous system, the duplicates evolve slower, but when the genes are expressed elsewhere, there's no difference in evolutionary rate. But if we compare duplicates which are expressed in the nervous system and duplicates which are expressed elsewhere, we find that the duplicates which are expressed elsewhere are expressed the same as the genes, the singleton genes, and the singleton genes which are expressed in the brain are expressed the same also as the gene expressed elsewhere. So we have only one category which is kept, which is really under strong selection, is genes which are kept in duplicate in the brain. This was a very complicated observation which took us a while to understand, but in the end, I will skip this one. We found a model which was published by another group on medical genetics and evolution, which is that if you have genes which duplicate, and then some mutations of these genes gives very gray phenotype, then it is very hard to mutate these genes. If you have a gene whose mutations are very deleterious, the natural selection removes the mutations. And so then it's very hard to lose the gene, because most mutations which make you lose the gene go through intermediate steps which are very deleterious. So genes whose function is very fragile, for example, genes which can be misfolded in the brain, are kept in duplicate under strong purifying selection on their sequence. Whereas genes which are either expressed elsewhere or less sensitive to misfolding, there's less selection on the protein, and you can lose the gene because there's a larger target of mutation to remove them. And so it's an interesting observation which now has been done by several groups and several species, that genome duplication tends to enrich your genome in what some people have called dangerous genes. Genes which are targets for mutation, which when the mutation happens is actually very negative. So here we have an effect of genome duplication, which is not to improve function, which is not to increase complexity, but actually to increase the number of targets for mutations, which give you problems, increase the targets for medical genetics if you want. So we find that many of the genes which are linked to genetic diseases are genes which were kept in duplication after the vertebrate genome duplication, because they're hard to lose. So the genome duplication duplicates everything. And because when you mutate these genes, you are sick, the mutation on the genes are eliminated by natural selection, so the genes are kept, and so they are there as a target for selection. So in a way, genome duplication makes us and even more fish, makes our genomes more fragile. So you see that it's not easy to put a direction to evolution, and maybe that's something I don't know that Christoph and Rob already said, but evolution does not have a direction, right? There's not improving things, it's just what happens. And maybe the selection short-term on the individual level to keep these two genes is going to make the species on the long-term more fragile, but that there's nothing to do about it. Natural selection does not see the future. So we enrich our genomes in dangerous genes, and they're kept by purifying selection. That's an interesting thing, that the intuitive thing from the 70s was that if you have duplicates kept, it's because they have a new function. So if they have a new function, the intuition is it was positive selection. You get 2 copies, one has a new function, so that's advantageous, and so you keep the 2 copies because one is advantageous. But here what we see repeatedly is that what we have is genes which are fragile, which are under purifying selection, were expressed in a complex organ, and which actually don't necessarily bring new function, but just it's hard to get rid of. So what we have is the duplication is a random event. It's a mutation, a macro mutation if you want, but it's a mutation. Then the genes are kept and duplicate by purifying selection, which prevents you from breaking things. But it is not fixed by positive selection. It's not advantageous. So you're going to get more and more genes without really an advantage for the individual or the species, at least short term. That's not what fixes it. Okay, I was afraid of being too long on this part, but I'm on time, so I'll be able to look at your questions. Cool. So an important take home on this is that if you look at paralogs and you want to look at the evolution of paralogs, you have to think that your set is always biased. So biased generation, I didn't speak so much about this because I spoke mostly about genome duplication. But if we look at other mechanisms of duplication, for example, tandem duplication can duplicate more easily short genes. Okay, because if you duplicate, when there's a duplication of a region of the genome, you should not think that the mechanism of duplication, this is a random mutation, it does not know where the gene starts and stops. It's going to cut randomly. So the shorter a gene is, the more chances that you duplicate the DNA region which has the whole gene and not half of the gene. Because if you duplicate half of the gene, it doesn't work. And there are other biases. For example, another mechanism of gene duplication is retroposition. So you have the gene transcribed to an RNA, and then there's a reverse transcriptase which translates, which makes this back to a DNA, which is integrated in the genome. Now this would happen more probably if there's more RNA for the gene. But for it to be relevant to evolution, it has to happen in the germinal line, at least in animals. It has to happen to genes. It has to integrate in a genome which we pass to the next generation. So if in my arm muscle, an RNA is reverse transcribed and integrated into the DNA, it does not affect my descendants. The only way it affects is if it's in the germline. So as a result, the genes are more expressed in the germline, spermatogenesis or oogenesis, will be more duplicated by reverse transcription. And because there are many more cell divisions in the spermatogenesis, then it's much more those which are expressed in spermatogenesis. So you have a bias to genes expressed in spermatogenesis duplicated by this mechanism. So you have a bias in the generation of duplicates, then you have bias in the retention, as I showed you, genes which are hard to lose. And then when you look at the evolution of the paralogs, you should think this always affects you. So you should always control for it. You should not just say, I'm comparing the expression or the function or the DNDS or the cellular localization of the duplicate genes to the non-duplicate genes in an organism and seeing the impact of duplication. I see both the impact of duplication, but also before that, the impact of retention of the genes, and before that, if it's not the whole genome duplication, the impact of the generation. So that's it for this part. It's a bit shorter. So now let's look at questions. Okay, so such high levels of gene duplication would mean that organisms in the same population would have different amounts of genes in pretty much every generation. Has anyone calculated the half-lives of gene duplication? So people have calculated the half-lives, but I haven't managed to find one consensus number. It's like depending a bit on organism studies. It's no, even for mutation, point mutation rate, it's relatively recent that we have a consensus number. There was the studies of families and of evolutionary divergence and of cell culture gave different numbers. But to answer your question on population, yes, in the one species where we do really a lot of population genomics is humans. And we find a lot of what we call copy number variants. And the copy number variant is a duplication which is not fixed, but is right now polymorphic in the population. So the equivalent for a duplication, so a substitution when it's not yet fixed, we call it a SNP. And a duplication, when it's not yet fixed, we call it the copy number variant. And we find copy number variants all over the human genome and all over human populations. So it's very common. And it's actually a big source of explaining human variation in various aspects. How do I know about the loss of about 85% after whole genome duplication? So that's with studies such as, I showed this simplistic, this cartoon figure, but it's been done in more detail. I will try to go back to the slide and share it. Wait. Okay, so you see my slides again. So when you have things like this, here you can count how many, you know that all the genes were duplicated because it was a whole genome duplication, and you can count how many now are duplicated with a chromosome position and age of the duplicates, which is consistent with the genome duplication. And so you can see which proportion of the genes now have a paralog consistent with the genome duplication, which proportion of genes do not. And that's how you get to about 15%, depending on organisms. But that's, for example, what you get typically in the teleost fish genome duplication, as well as in yeast. 15% approximately of the genes have this pattern of having both kept in duplicate, and 85% having this kind of pattern. Depending on the age of the duplicate, you get anything from 50% to 10% of the genes kept in duplicate. Usually, once you reach 10, 15%, it stops going down because these genes have been fixed for various reasons, either because they've created a part of a network where you cannot break things by removing a part, or because they got a new function eventually, something. So in all the example studies, what could be the impact of absence of information, such as expressed absent might be a third level category, information unknown. How can this be controlled? Well, that's where it's important, depending on the type of study you do, to indeed have the proper control. So if you're doing, for example, an enrichment table, you should only, if I'm looking at genes expressed in a certain circumstance, for example, in the brain, my reference should be genes for which I have expression. For example, that table I showed you, I didn't go in detail, but that table specifically is only from in situ hybridization data, which has the highest level of resolution zebrafish. And for this, our reference was the genes which have been studied by in situ hybridization in zebrafish. But this will depend. You should always think for every study, case by case, what are my potential biases and what are my controls? It's a very good question. You should always control. For DNDS, are synonymous, non-synonymous mutations always identified by comparing the sequence of interest to an outgroup or common ancestor? Yes, always by comparing to an outgroup. So to calculate DNDS or KAKS, you always need at least two sequences. But if I'm comparing paralogs between themselves, I need to compare them to an ortholog. And if I go back to a slide there, actually, I'll get back to the right slide and then I will share it. Yeah, okay, so sharing the slide. Okay, so here, that's why for this study, so this is 2004, there was still, it's not 2000, but there were not that many genomes available. But it was important to have these two insects here so that you could calculate DNDS between the two. You couldn't just have one insect and calculate DNDS on its branch. DNDS always has to be in a comparison. So now we have phylogenetic models, maximum likelihood models, which are more complex than the original ones were just comparing a pair of sequences, but still you need, for the phylogenetic models, you need at least 4 sequences actually to calculate DNDS on the different branches. So yeah, you have to be able to compare something always. It's funny, every time I leave Zoom and come back, I have to scroll down to the latest questions. Is whole genome duplication linked to speciation? If you have the answer, call me. Seriously, it's been a big hypothesis for many years. For example, when genome duplication was discovered in teleost fishes is the most diverse species group of vertebrates. So it's about the same age as birds, which there are about 8,000 species. And there are about 25,000 species of teleost fishes, which is about half of all vertebrates. So you take all vertebrates, including sharks and birds and mammals and lizards and whatnot. Half of these species are fishes, teleost fish, and they had a whole genome duplication. So this was a hypothesis which proposed in the literature. And also there were some anecdotal cases in plants of clades which had a hybridization or a genome duplication at their origin, which also suggested this. But then it's been extremely difficult to test because you can always find an example. So I look at the species group, I find the genome duplication, cool. But now how do I control this? How do I make an unbiased study? Because we don't sequence random genomes, so our knowledge about genome duplication is not random. And it's very difficult to estimate also what happened in time. So if a genome duplication happened, for example, in teleost fishes 300 million years ago, but the big divergence of teleost fishes was, I don't know exactly, 100 million years ago or something, that's a huge lag. It seems difficult to explain like this. And we know that groups like beetles are very diverse without genome duplication. And we know that some groups have genome duplication like sturgeons and went nowhere in number of species. There's a handful. So there's a whole debate among people who are specialized in modeling speciation and extinction rates, who regularly publish with a new and improved model and find the opposite of the other team and argue. So there are some specialists of speciation rates who think that genome duplication does increase speciation rates. There are some who think it does not, but it decreases extinction rates. And there are others who think it has no impact. I don't know. I can tell you, we tried to study this in the case of fishes. What we found is that we could find no relation between the timing of the duplications and the timing of speciations in the fossil record. So we analyzed together genomes and the fossil record, which was kind of fun, but we found no pattern, which was kind of disappointing and made us a +1 paper, which is what happens in such cases. So yeah, I don't know. I'd like to know. Are there questions? Can the assistants ping me on Zoom? Are there questions I should look in the Google Doc? No, okay. So I suggest we make a 5-minute break and start the next part a bit early because I never know if I'm going to be a bit faster, a bit slower. Okay, so 5-minute break, coffee, bathroom, and I start again, okay? See you. Welcome back. So I hope you all had time to have a small break. I'm sorry, I'm such a slave driver, not giving you any breaks. I'm awful. So I'm putting directly on the screen the next WooClub poll. This way you can look at this. No love for paralogs for now. I feel the influence of Rob's talk here. I think he mentioned phylogeny for orthologs. Okay, so I'll leave the poll up, but it's pretty clear that what's winning is first that you need orthologs for the species phylogeny. Secondarily, that only orthologs can be compared or only orthologs share function, which is kind of two ways of saying more or less the same thing. And no one thinks paralogs are interesting, which makes me very sad because I started using ortholog databases to find the paralogs. There's something also about bioinformatics, you can torture it to make you give you something else. You can take a database of orthologs to get the paralogs because they're the ones which are not orthologs. So I will stop sharing this. You can continue voting. And I will share my slides. Okay, so part 3 and last, evolution after duplication. So now that we know that we can characterize the function of the genes somehow, that we know that they duplicate not unfrequently, that this duplication and retention are biased that we have to take into account, can we all the same see, okay, how do they evolve after duplication? And so we just did this, and most of you think orthologs are important for species trees. I bet that's because Rob said so yesterday. I bet most of you don't do species trees in practice when you do comparative genomics. I should make a new poll on this. Do you actually do species trees in your research? You know, the main reason most people want to distinguish orthologs and paralogs is that they want to annotate function, and they think that function can only be annotated by orthologs. And this is a rather old idea. So Susumu Ono, I mentioned him before. He was the first to suggest that genome duplications were important in vertebrate and mammal evolution. He had a rather, let's say today, outdated view of evolution. He thought that mammals are more complex and superior, and that this could not happen just by point mutations, but you needed large-scale macro mutations of genome duplication to create the new material to make this complexity. And he has this very famous quote, natural selection merely modified while redundancy created. If you try to really think about this and put in equations, it doesn't mean much, but it corresponds to a kind of intuition that when you have redundancy, when you have more copies of the genes, then you can get new functions. And this is a very strong intuition. And for many years, so the terms ortholog and paralogs were invented in 1970. And I did my PhD in the 1990s, and I was told always that you need to find orthologs because orthologs have the same function and paralogs change function because after duplication, you get new functions. And this was not tested for a very long time. And only recently, in the last 10 years, have people started investigating this. And although I'm pretty proud to have published a paper in 2009 before anyone else asking the question, I'm not so good at marketing apparently, and someone else invented the good, cool term, which is the ortholog conjecture. So that's how we call it. The ortholog conjecture is the idea that orthologs keep function and paralogs don't. So you can think of this hypothesis or conjecture in a more phylogenetic way or a more pairwise comparison way. So if I think of it in a phylogenetic way, here I have a gene tree, okay? Here are the species. So I have species 1, species 2, species 3, species 4, species 5, species 6, species 4 again, species 5 again, species 6 again, species 6 yet again. Why there are species again? Because I had duplications, and this is a gene tree. So in a gene tree, you can find several times the same species, because after the duplication, you get 2 copies of the gene, and so you have the species twice. And here, under the ortholog conjecture, let's assume that every time that the function evolves very slowly after speciation, almost not. So the color here represents a kind of quantitative measure of function, like you would say an enzyme activity or a level of expression, something you can quantify. And the letter is more a function that you would describe in words such as gene ontology term. So let's say that here, after the duplication, I have a jump. So I have a huge quantitative change. I go from these variant reds to these variant blues, or I have a difference in the function I describe. Say I catalyze a different reaction, I bind a different ligand, and so the function goes from A to B. And here at this duplication, it goes from B to C or to a totally different color again. And so in this view, function can evolve very slowly. Most of the time when there's duplication, it jumps, and so the orthologs without duplication at all have the same function. When I have one too many orthologs, such as between this species 3 and the species 4, there's this one and that one, then there's one ortholog which kept the function, one which didn't. So that's a phylogenetic way of seeing it. And an alternative, if this hypothesis is not correct, is that during evolution, I have duplications, and in an unrelated manner, I have at random paces in evolution, shifts in function, but the two are not related. And so this can mean that I can have, as here, 2 orthologs which have close functions, but I can also have paralogs here which have close functions, or I can have orthologs like here which have very different functions. Now the thing is here, this is important, is that if I compare few species, I might not see the difference between these 2 patterns. So if I'm only looking inside one species at the paralogs within the species, if these paralogs have different functions, for example, I look in species 4, here I have this paralog and that paralog. Here they have different functions, A and B. Here they have different functions, A and B. So the fact that I find paralogs inside one species, yeast paralogs, human paralogs, Arabidopsis paralogs, which have different functions, does not tell me that the ortholog conjecture is true. It does not tell me that the duplication created the functional change. Just tells me, since the duplication, enough evolution happened that at some point there was a change in function. But maybe it happened related to the duplication, like here, or maybe it happened unrelated, like here. And another way of seeing, so to test, in this case, I need to make phylogenies and have some kind of measure of function that I can map on phylogenies. Another way of seeing it is to compare pairs of genes. And these pairs of genes are either pairs of orthologs or pairs of paralogs, and they have a certain age, a bit like in the graph I showed you at the beginning of the previous hour, where we had the duplicates which had different ages measured by synonymous change. So I can use some proxy like this, either a phylogenetic estimate of aging or synonymous variation or something like that, to estimate the age of duplicates. And now under the ortholog conjecture, at birth, I mean, just after the speciation, orthologs should have very high functional similarity. Oops, sorry. So here I'm measuring some measure of similarity of function. Maybe it's that they have the same expression, maybe they're the same gene ontology term, maybe they have the same interaction partners, some measure that they have the same function. And the orthologs at speciation are almost the same, whereas the paralogs already at the duplication might be different because maybe they duplicate to a different genomic environment or something. And then as time passes, evolution happens, so function diverges, so the functional similarity decreases, but always for given age, the orthologs are more similar in function than the paralogs. So here you allow evolution to happen for both, but you say it happens faster and steeper for paralogs. This is the alternative. If there's no ortholog conjecture, they both diverge in the same way with time. And again, depending on the experimental design you have, you might see different things. If, for example, I'm comparing human mouse orthologs and then human paralogs which come from vertebrate genome duplication, then the human mouse orthologs have this age, and so this functional similarity, whereas the vertebrate genome duplication is much older, so the paralogs have this age and this functional similarity. So under both models, in that case, if the paralogs are older than the orthologs, obviously the paralogs are more divergent in function. So you have to be very careful, and that's something we saw when we started studying this in 2008-9, is that you have to be very careful of your study design. And a lot of the early papers saying that duplicates diverge, early I mean papers from 2000 to 2010, they just compared paralogs within one species, and they found they were different, and they said, hey, see the impact of duplication. But in fact, any model where evolution is non-zero will have the paralogs a bit different. The question is, are they specifically different? Are orthologs more similar and paralogs more different? That's the question. And so this question, despite, I would like to say that when in 2009 I published a paper saying, hey, we should study this, it had a huge impact, but the sad truth is no one read it at the time. And what happened is that in 2011, a study was published giving apparently experimental support for the ortholog conjecture being false. And this study had a huge impact, and then people read our old paper and everyone else continued studying it. So this paper from 2011 by the group of Matthew Hahn, and so they compared the similarity of gene ontology annotations as well as the similarity of gene expression measured at the time by microarrays between orthologs and paralogs in human and mouse, according to the divergence between the genes, which gives you a measure of age. And what you have here, you have, so this is similar to the theoretical graph I had here, but this with real data. It's the gene ontology annotation similarity. And here you have in red the orthologs all the way down here, or down here. So not very similar. And up here you have in paralogs, which are paralogs which duplicated after the speciation between human and mouse, all the way up here. And here within species out paralogs, so duplicates which are older than the speciation human mouse, for example, from the vertebrate genome duplication, but where both copies were studied in the same species. And what you see in these graphs is that you have not only not the ortholog conjecture, you have the opposite. The genes which have the most similar function are the paralogs. And the genes which have the most different function are the orthologs. So don't start taking too many notes on this because it's not correct, but it's an interesting error. There are two types of errors, errors which teach us something and errors which don't teach us something. And as a scientist, you want to make the errors which teach you something, because if your aim is to never make mistakes, I have news for you, you will make mistakes. So will these be fécond mistakes? And so actually this was very interesting to Christophe Decimo, and I started the collaboration in 2009 after we published that maybe the ortholog conjecture should be tested, and they published a paper using gene ontology annotations to test orthology. So we said, to test orthology using gene ontology, let's do the opposite, yes? Use orthology and gene ontology to test the evolution of functions. So we did this, and we got almost exactly the same graphs. We didn't choose the same colors, but apart from that, we had the same graphs. But we didn't publish it at the same time as they did because we found there was an error in this. There was a bias, and this is actually an interesting bias because it shows how we have to be very careful in comparative genomics and what we compare and how. So here I'm going to summarize a rather complicated bias unraveling in one slide, so bear with me. If you think of the labs which are experimental, they usually study one species. So you don't have many labs which study the orthologs of one gene in mouse, human, zebrafish, Drosophila melanogaster, C. elegans, Arabidopsis, yeast, and so on. You have a lab which will study some process and the gene families involved in it in zebrafish or in fly or in Arabidopsis. And actually these labs then are part of communities which do the same type of experiments, use the same vocabulary to describe things, exchange students and postdocs. And so what we found is that if two genes were published from the same paper, so have the annotation from the gene ontology from the same paper, the similarity of function here is very high, and it's not different between orthologs and paralogs. Oops, sorry. If the gene ontology comes from studies published in different papers but which share authors, so the same labs basically, the similarity is much lower. And again, not a very striking difference in orthologs and paralogs. If I only take gene ontology annotations which come from papers with no common author, then the similarity is even lower. But here I have a difference. This is the color of the orthologs in this case. The one-to-one orthologs are significantly above all along. And now this is interesting because same species paralogs, so paralogs from the same species, say Hoxay1, Hoxay2, Hoxay3 of zebrafish, would tend to be studied in the same papers much more often. Okay, 47% of same species paralogs annotations come from the same paper. But orthologs much less often, only 1% of one-to-one ortholog annotations in the gene ontology come from the same paper. So what we have is that when you study things in the same paper, you do the same type of experiments, ask the same questions, answer in the same way, using the same terminology, the same vocabulary. And then when there's the bicuration, remember bicuration this morning? The bicurators read the papers, they don't do the experiments. So they read a paper, and this paper on gene A and gene B finds the similar stuff with the same terminology described in the same way, with the same type of figures, same type of results, so they give it the same gene ontology term. With some other papers, different papers on orthologs, one did some set of experiments, the other did a different set of experiments, and they described them differently. So since the gene ontology is capturing words in the end, which represent concepts, but they're reading the words, they're going to capture different things. And so we find that the orthologs tend to be captured with different terms in the gene ontology because they're studied by different people. And the paralogs, especially the in-species paralog, tend to be studied by the same people and so have the same gene ontology annotations. And so that explains actually this result here, that even the old orthologs within species out paralogs, the old paralogs, sorry, have more similar function here than the orthologs because they were studied by the same people. This is not biology. This is actually very interesting, and I mean, I have a 45-minute talk just on this part, but I'm not bothering you with it today, but you can really disentangle what in the gene ontology allows us to do comparative genomics or not. And it's actually very tricky to do comparative genomics with the gene ontology. It was not meant for that. It's not meant to be compared. So you have to be very careful if you do compare. And these biases are at all kinds of levels. So what we found at that point in 2012 is a kind of tentative answer that if you correct for a lot of biases, and here I showed you one bias, but actually this gene ontology annotation had at least 4 major biases. You get this difference here, which on the one hand is significant statistically because we have a lot of genes. This is numbers here. You get, I don't know if you can read, 160,000, okay? So it's significant. On the other hand, the effect size, which is what should interest us as biologists, is very small. What we want to know is not just, is the p-value small? Because in genomics, scoop, you always get small p-values because you have big numbers. But that's boring. What you want is to get big effect size, something which actually explains a lot of biology. And here the difference is tiny. So if this is the final word, it means, yes, orthologs are a bit more similar than paralogs, but really you shouldn't care. So we looked for other ways to do this, and I told you the gene ontology is not very well adapted. So us and many groups tried to look at gene expression, because gene expression, I showed you this morning, can be compared between species, and it's kind of objective measure of function. So it only captures a part of function, but you can characterize it much more objectively. The problem is that there's a lot of issues in comparing also gene expression between species. So if you correlate, then you have biases on the more specific genes have higher correlations. I'm not going to go into detail, but there are many issues with this, and there were quite a few papers comparing gene expression between species, which each said it was conclusive, but if you put them together, it was not conclusive. So we decided to look at gene expression a different way, not just the level of gene expression, but the specificity. So the idea was that one measure of function is either a gene is housekeeping, it's ubiquitous, it's expressed everywhere at the same level, or it is specific. For example, it's a gene expressed highly in the brain and lowly elsewhere, or highly in the muscle and lowly elsewhere. So it has a tissue-specific function. And this describes an only very small part of function, but the advantages that we showed in some boring bioinformatics paper, that it is very robust. So you can measure different ways using different data sets, and you always get the same conclusion. And that's very reassuring, because we want to compare different species, and in different species you have different experiments done by different labs with different techniques. So you need a measure which is robust. Otherwise, you're just measuring the differences between labs. There's a famous paper a few years ago where they said that actually orthologs totally don't have the same gene expression in human and mouse. And there was a follow-up paper by another lab said, well, actually you use different generation Illumina machines for the human and mouse, and that explains everything you see. So you see, you have to be very careful. So tissue specificity, we showed that it was very robust. And so then we compared the tissue specificity between orthologs and between paralogs. And we got this. So here you have one point is a comparison. It's one data set of expression compared between either two species for orthologs or the paralogs in a species for a given age. And we did the age, not this time by sequence divergence, but by the phylogeny. So you can make the tree, the phylogeny. If I show you, this is something, in fact, people often have issues with, so I'll show you how it's done. If you have a phylogeny like this, I can say this duplication happened after the speciation and before that one, just from the tree shape, okay? So I can date this duplication at this point, somewhere on this branch. So that's how we did. So it's a more robust way of aging, dating. And so what we get is here the orthologs have very similar tissue specificity. So if you take recent ones, like here's human chimpanzee and human gorilla, there's an almost perfect correlation of tissue specificity between orthologs. And as the species are more divergent, this goes down, which is kind of reassuring on the fact that evolution happens. Because anything which is a flat line is kind of disturbing when you look at evolution. You think the more time passes, the more it will change. And the paralogs also go down, but more importantly, they're always under the orthologs here. So here I'm comparing all the paralogs inside human. We also did it centering on different species, but here it's centered on human. And you find that the paralogs have a much lower correlation. They don't have a zero correlation, okay? Paralogs are not random pairs of genes. They are genes which share a lot of features, so they do correlate, but much less than orthologs. So when we published this, we were very happy because we thought that now we have answered the question. We had this question since 2009. Are orthologs more similar in function than paralogs? And my secret hope was that the answer would be no, because it's much cooler to overturn what everyone thinks than to confirm it. But okay, I can settle for second best, confirming it. And this looked very robust, and I was very happy. But science doesn't stop just like that. And actually, I told you this is pair the phylogenetic view. And a few years later, so that was 2016, this is 2018, 2 years ago, Kassedun, a specialist of phylogeny in Yale, published this paper. He was nice, he gave me a head warning of a few months, saying if you do the pairwise comparisons, you're comparing things which are not independent, because it's a tree, actually, the evolution. See here, you have twice species C because there's a duplication. So here you have the duplication, you have the paralogs. So the comparisons here are all mixed up. What you should do is take the tree and look at the evolution on the duplication branches and on the speciation branches. And there are actually tools developed for this since the 80s for the evolution of phenotypes on trees to calculate how fast phenotypes evolve on phylogenies. Notably, phylogenetic independent contrast, PIC. Phylogenetic independent contrast is simply how much the trait diverges on the branch relative to how much we expect from the branch length, which is expressed as a variance. I'm not going to go into the math here, but basically you transform the branch length into a variance that, and then you look at how much the phenotype changes relative to this. And then you can compare these normalized changes. And what's interesting is that you can reconstruct the ancestral states at the points here of the phenotype. And so you can calculate also at the internal branches here. Oops, sorry. When I move the mouse, it tends to change slide. And so you can compare now the PIC, the phylogenetic independent contrast, for the duplication branches and the speciation branches. And now you can have a proper test of the ortholog conjecture. And so what they found is that when they took real data, so they reproduced our analysis with only part of our data for various reasons, they found the same result as us, which is kind of reassuring and shows that we did reproducible science. So all our code was shared publicly, so they were able to just pull our code and rerun it, and they thanked us for us, and I think it's natural. And I really encourage you to share your code, because it allows such things to go much smoother and not to fight with people, but to work together, because then they share their code and we're able to build on it further. So really do open science, share your code, we'll all get further together. But when they did phylogenetic independent contrast, I find this figure very unclear, but what's important, they compared by Wilcoxon test the phylogenetic independent contrasts between the duplication branch and the speciation branch, and they didn't find any difference on the real data. And now they did simulations. If they simulated data under the ortholog conjecture, they found a difference like we found in the real data on the pairwise comparison, and they found a difference on the phylogenetic independent contrast. But when they simulated without ortholog conjecture, they still found a difference with the pairwise comparisons, which implies that they're biased. They find a difference even when there is none, because when you simulate, you know what you're doing. You know there is no difference. Whereas the phylogenetic independent contrast managed to find no difference. So they argued from this that actually when you correct for the phylogeny, there is no difference between the functional evolution of orthologs and paralogs. Now, I had a number of issues with this study. Not that they contradicted me. As I told you, I was actually hoping to find the ortholog conjecture first because it was more fun. But because here they treated the branch length as something very reliable. This is what we normalize by. But we know that duplication and evolutionary rate of genes are not independent. We know that duplicate genes are not a random set of genes relative to their evolutionary rate, which is going to change the branch length, because the branch length is the product of the evolutionary rate times the evolutionary time which passed. And also that after duplication, we know that there's weaker purifying selection for letter Y, so genes accelerate, so branches are longer. And so we revisited this, and the details in this bioarchive preprint, which I really don't have time to show you all the details because it gets quite technical. But basically, if you correct the trees for all the biases, which in fact are in trees which have both speciations and duplications, then you recover a difference in phylogenetic independent contrast, which is larger for the duplication branches. So in fact, if we correct for phylogeny, taking into account that phylogeny of a gene tree with duplications and speciations is not the same as phylogeny of a pure species tree, then we do recover the ortholog conjecture. I wouldn't say this is the end of the story, because obviously every time I thought it was the end, it wasn't, and the same for my colleagues who say the opposite, but it shows that you have to be very careful with these methods when you have gene trees which mix duplication and speciation, because there's a lot of biases to take into account. Just to give an example, another bias is that you can date, you need absolute dates for these methods. You can date the nodes which are speciations from fossils. We cannot date duplications from fossils, we have to estimate them. So it's not the same, and this introduces also a bias. And this is still not the final word, but a recent paper I found very interesting on the ortholog conjecture, and in general, a really cool paper if you have the opportunity to read it. They knocked out genes in yeast and replaced them with the human ortholog. And so if they recover the function, so if you knock out the gene, you lose some function. If you put back the gene, you recover the function. Now, instead of putting back the gene, I put back the ortholog from human in yeast. And if you have one-to-one orthologs, you replace in about half, it works. But if you have one-to-many, so there was a duplication somewhere in the branch, in the human branch, that could be anywhere, ancestral to animals, to vertebrates, human specific, whatever, they don't know. If you have a duplication here, then only in a quarter of the cases you can replace the function. And if on both sides you had duplications, then only in a small proportion. So it means that there's clearly something which happens during the evolution of the function after duplication, since it's much easier to replace. And here we have a full measure of function, right? The yeast lives happily or doesn't. So this, I would not say, is not a real test of the ortholog conjecture, because you could immediately see that they don't control for differences between generation, retention, and evolution after duplication. But it's a hint that yes, one-to-one orthologs do have more similar function than other genes. So I would like to know what you think of all this. Do you believe the ortholog conjecture or not? So I'm going to go to my WooClap. Where's WooClap? To go to the next question. There. So vote from what I told you. Do you believe the ortholog conjecture? The race is on, and it was very close at the beginning, but now there's a clear majority coming up for the ortholog conjecture, which I suppose is reassuring to the speakers of the previous 2 days whose full-time activity is to find orthologs. My specialty is working on duplicates of paralogs, so I'm a bit, I have a different perspective on this. Although I've spoken with specialists of duplications who clearly believe strongly in the ortholog conjecture, because I think the only way you get new functions is by duplication. So, okay, so let's say that after all I said, I don't know how biased or unbiased I was. It's impossible to be totally unbiased. We get 80% of you who believe in the ortholog conjecture and 20% who are unconvinced. Okay. So let's go back to my slides. Next slide. So I think one thing I take home from this, I don't take home whether the ortholog conjecture is true or not at this point. I think it's probably true because I've looked at this data really a lot. And most of the time when you're careful, you find the ortholog conjecture. But what I find really interesting is that it's really hard to prove something which is supposed to be obvious. When I was a student, when I was doing my PhD in the 90s, it was obvious that orthologs have the same function. And the first time I presented in a meeting, in a conference in late 2000s, 2008, I think. Hey, in fact, we should ask this question. We don't know. People were shocked. People who work on orthologs were telling me, no way. You cannot even ask this question. It makes no sense. And actually, when you ask it, it's surprisingly hard. For something which should be obvious, it's surprisingly hard to prove it. And this, I think, tells us a lot about where we are in comparative genomics when we want to go beyond genes to function. If we are not able to answer this question, then it means it's very hard for us in general to answer any question about the evolution of function. That's why I spend time on this, not just because I was involved in it, but because I think it's an interesting window into the limitations of all our methods for doing functional comparative genomics. And this is a little table I published recently, summarizing a bit the ways you can get information which can be compared or not, like from the gene ontology or expression or whatever. If you're comparing two genes, whether they're orthologs or paralogs, and the function was transferred by orthology or by homology, you're going to see they have the same function, but it doesn't mean anything because you transferred it by homology. And this looks ridiculous when I put it like this, but there are papers using, for example, the enzyme committee nomenclature. So you know those little numbers between other enzymes, 1.12.4.1. These numbers are overwhelmingly transferred by homology, and there's no annotation allowing you to know how they transferred. And so, of course, they find similar functions between orthologs because they transfer them between orthologs. But the same goes for a lot of function annotations. If you have function X in one species and function Y in the other species, but because you did different experiments, the functions look different, but no, just you did different experiments, so you cannot directly compare. That was the problem of the different papers and same papers. Now, if we did the same experiment and we get the same function, this gives us information about evolutionary conservation. And the easiest case of this, obviously, expression, I do RNA-seq, I have the same expression, then it's conserved. And if I do the same experiment, I have different function, I do RNA-seq, I have changed an expression, this tells us evolutionary change. Just to tell you that this can be tricky, for example, there have been papers comparing the essentiality of genes between orthologs. So an essential gene is a gene, if you knock it out, you're dead or sterile. So you do not, your fitness is zero in evolutionary terms. And so people have compared the knockout phenotypes between, say, zebrafish and mouse, or between mouse and human medical genetics results. But then when you dig deeper, I tried to do this and we never published because it becomes very complicated, because knockouts are very rarely clean. I knock out the gene from one end to the other exactly. You knock out part of the gene, you make you put the stop codon in the middle, you remove part of the promoter, you have a conditioned knockout only in some tissues. When you look at all this, it's almost never the same experiment which was done. So you cannot really compare. So it becomes very, very difficult to compare the function of orthologs or paralogs when actually the experiments were different. Another little poll. I will go to WooClap to activate it. There. So this one should be fast because we've been speaking about this a lot. I see I'm running a bit late on time, unlike previously where I was early on time, so I will go back to my slides while you guys can vote in the background, okay? So now we're going to come to the most interesting part at the end. No, it's not true. Everything is interesting. Love all comparative genomics. Evolution after duplication. So here's a little figure I did to summarize all the different levels at which function and duplication can interplay. So you have the different types of mutations, the fixation of the duplicates after. So you should think, by the way, that duplication is a type of mutation. Even genome duplication is a type of mutation. Because sometimes people will say mutations can't create new functions, but duplications can. But that's false, because duplication is a type of mutation. So then you have the fixation we saw before, we saw the retention before. This is population genomics, and we did not speak about population genomics in this course, but when we spoke, for example, about the variation in populations of copy number variants, it's at this level. Then we have this bias by gene function, which retains different duplicates according to their function. But then after they are kept, they can evolve their function in different ways. And so the most easy way here is dosage. So now I had one gene with two patterns of expression from different promoters. I had one originally, and now I have two, and the two are kept and expressed, and so I have twice the amount of product. And this happens. For example, ribosomal proteins, you have arrays of duplicate genes which express the same protein because you need a lot of ribosomal protein. But, oops, sorry. The hypothesis which, where did I go? There. The hypothesis which was the most popular when people started speaking about gene duplication was what is now called neo-functionalization. So you have two copies of the gene. One is under purifying selection to keep the ancestral function, which is still needed. So the other is no longer under strong selection. So mutations, including large effect mutations, can get fixed. And so this allows the evolution of new functions, whether it's new promoters, new domains of expression, or new protein functions. And that's neo-functionalization. And in the early 2000s, Mike Lynch, the same person who made this frequency of duplicates I showed you earlier this morning, proposed the third model to explain why we found so many duplicates. Because if this is the only way, and that was for a long time what we thought to be the only way, it should be very rare to keep 2 genes, because the probability of a mutation which breaks something is much higher than the probability of the mutation which improves something. Okay? If you give your computer code to someone you don't know and say change what you want randomly, chances that he improves it are much lower than chances that he breaks something. Right? So random change usually don't improve things. And the beauty of the subfunctionalization model is to say, if I have several subfunctions which are independent, here, for example, it's domains of expression, but it could be protein interaction partners, it could be subcellular localization, anything you can imagine to be separate. It's easy to have a mutation which loses 1, because that's breaking something. And now if one copy loses one subfunction, the other copy loses the other subfunction, I have no gain of function, but now I need to keep both genes to keep the ancestral function. To have, for example, these 2 expression patterns, these 2 domains of expression, I need to have the 2 copies. And now I'll keep both by purifying selection, because I want to keep the ancestral function, which was useful to the organism, without any gain. And so this model was very powerful and interesting when it was published in 2001, because the first model to say we can complexify genomes without any gain. This comes back to the question I asked you earlier, whether genome evolution is related to some concept of higher animals. We can actually increase enormously the complexity of a genome without any gain of function. It becomes a kind of a Rube Goldberg machine. If you know Rube Goldberg machines, is when you have the ball falls and breaks the balloon, which overthrows the water, which wets the cat, which jumps onto the ladder, which makes the book fall, and in the end serves you coffee or something. So it's useless. But if you remove part, once you make something so complicated, if you remove part, it doesn't work anymore. So duplication can create that actually through subfunctionalization. And so a bit like the ortholog conjecture, once you have these 2 hypotheses, you want to test them to see in reality, in practice, I mean, I can make some theoretical model, and on a theoretical model, this is frequent and this is rare, and that's what they did in the early 2000s. But the theoretical model has a lot of assumptions, and we don't actually know what really happens. So what really happens? So it's been studied in several ways. And one way is to look at natural selection, so DNDS on the sequences. And just to say what were the expectations of selection on the different models. So here, this is a bit complex, but bear with me. Here you have the gene frequency. So gene 1 is always there at 100% frequency in the population. And gene 2 appears at the duplication in one individual, increases in frequency, and then gets fixed. And now I have 2 duplicates. So now it's at 100% in the population. Everyone now has 2 genes. And neo-functionalization means that at one point, so here, nothing is happening. Here I have the 2 genes and it's drifting. But then I have a mutation in black here, which gives a new function to this copy. And now positive selection, see the curve increases slope tremendously. Positive selection is pushing for the fixation of this gene because I have a new positive function. Positive selection is also pushing to keep this one because you need both. And then you have purifying selection, which keeps both. And this is subfunctionalization. I have a gene, I have a duplication, I'm drifting. Now I lose a function in one of the 2 genes, but they're still redundant on part of the function, so still drifting. But now I have a mutation on the other gene, the other subfunction, and now I have purifying selection keeping both, because I need both to make my function. And the big difference between these 2 graphs is this red part here. In this model, there is positive selection. In the subfunctionalization model, it's neutral. There is no positive selection to fix mutations. This has been tested in several ways and never convincingly, so I don't have a convincing result to show you, but I can show you a little bit of evidence. So one thing we see repeatedly is that duplicates evolve asymmetrically. So you have 2 duplicates and one evolves fast and one evolves slower. And this has been interpreted as evidence for neo-functionalization, because here, one of them gains a new function and changes, and here not. But then, and here they have, when you cartoon this, you always put, each has one loss of function. And that's also what I put in my cartoon earlier. Here, they each have one loss of function, right? So you always draw it symmetric, whereas here it's asymmetric. But actually, there's no reason for that. There's no theory which said it should be asymmetric or symmetric. So you can have asymmetry under subfunctionalization also. So asymmetry, although there's been quite a few papers which said it was indicative of neo-functionalization, that's not necessarily the case. So what you need to decide whether it's subfunctionalization or neo-functionalization is to know what was the outgroup state, because a big difference between neo-functionalization and subfunctionalization is that together they describe the function of the outgroup of the ancestor, or one has the function of the ancestor, the other has a new function. Now we don't have a time machine because the physicists still haven't given us a time machine. And if you have any physicist friends, you can complain to them. But we have outgroups, which is second best. And that we have thanks to comparative genomics and orthologs. So let's rejoice. We should compare our duplicate genes to an outgroup not duplicated and see if one copy is like the outgroup in function and the other is different. This looks like neo-functionalization. If the two paralogs are a bit different and together they describe the function of the outgroup, that looks like subfunctionalization. And I don't have a result of mine on this because I've been trying to test this for years, and every time I try to start testing it, there's new results on the ortholog conjecture, and I go back to that. So it's still my plans for next year, but it's been my plan for next year for 6-7 years. But other people have worked on it in the meantime. So here, for example, you have RNA-seq human mouse, where they looked at the expression of the duplicates which are human-specific or mouse-specific, comparing to the outgroup and the other species. And for example, here, here they don't have an outgroup, sorry, my bad. So you cannot tell. So this is an example of asymmetry. Here you have these two genes which have different expression. See, this gene is expressed highly in the lung, the kidney, the liver, and the testis. And the paralog is also expressed in the testis, but highly in the heart, where this one wasn't, highly in the cortex, where this one wasn't. And here I have a gene which is highly expressed in one copy and lowly in the other. And actually, this is quite frequent. It's never in our models, but it's quite common. So this kind of study is interesting, but doesn't really allow us to choose. So we need outgroups, and this is where the fish genome duplications are very interesting, because in fishes we have not only genome duplications, which means a lot of genes which are duplicated with the same age, so we can compare them, but we also have outgroups. For one thing, a nice outgroup to all fishes is human and mouse, where we have a lot of data. So when I work on fishes, I treat human and mouse as the outgroup, which is very convenient because there is a lot of data. And we also have extra genome duplication in salmonids, and salmonids have an outgroup all the other fish for which we have data, notably zebrafish. And I wish I could tell you I'm going to give you the final answer here, but it's not the case. So this figure is a bit complicated, but what's important is that here, if you compare, so here what they did, they sequenced the gar, and the gar, if you go back here, is a fish which diverged by speciation from other fishes before the teleost genome duplication. So it's an outgroup to the teleost genome duplication. So all the genes are duplicated in all teleost fishes from the genome duplication, a single copy in the gar. And so they did the genome and the transcriptome of the gar and compared expression. And so we have genes like this, where you have the gar in gray and one paralog in zebrafish, which is expressed in the same place as so one paralog here in red is expressed in the same place as the outgroup, the gar, and the other paralog in blue is expressed in a different place. So that looks like neo-functionalization. And you can do the same thing when you have the MEDACA, so it allows you a biological replicate, the different fish also teleost, and they have the same pattern. But you also have patterns like this, where the gar gene is expressed in the brain and the heart, and one copy of the gene duplication, the onolog, in zebrafish expressed in the brain, and the other in the heart. So this really looks like subfunctionalization. And when they counted the two types, they had much more subfunctionalizations than neo-functionalizations. They also verified this by correlating the levels of expression. So here, for example, you see that here you have the one copy in the zebrafish is lowly correlated to the gar outgroup, The other copy is also lowly correlated to the GAR outgroup, but if I sum their expression, so on subfunctionalization, I'm going to assume that the sum of the two corresponds to the outgroup, then I have a high correlation to the GAR, similar to what I get for singletons. So this is for the genes which have a pattern like subfunctionalization, whereas those which have a pattern like neofunctionalization, the duplicates differ from the singleton, so it's not the same. And you repeat again this in zebrafish and medaka with slightly different scales. And here they found evidence for overall subfunctionalization. That's very nice. It's a lot of data. It would be nice if we would finalize the question, but the same year, another group studied the salmonid genome duplication. So they sequenced the genome of a salmon which had a duplication, the salmon salar, the usual salmon, and pike, which is closely related to salmonids but didn't have the genome duplication. And they compared the gene expressions like this. So you have gene expression, every column is a tissue, a sample, and every line is a gene. And so you see that when you have a gene, which, for example, here, all these genes are highly expressed in the brain for one copy in salmonella, in the salmon, highly expressed in the brain for the outgroup in the pike, but much more lowly expressed in the brain for the other copy in the salmon. And on the other hand, they had high expression in all kinds of other places which was not present, neither in the outgroup, nor in the first copy. So this goes in the direction of saying that we have neofunctionalization. We have repeatedly genes which have one copy which is like the outgroup and the other copy which is different. So these two groups have fought by different papers. They use different methods, different metrics. So there's a paper by the salmonid group which says if we reanalyze everything, it's clearly neofunctionalization, and a paper by the GAR group which says if we reanalyze everything, it's clearly subfunctionalization. At this point, I would say I don't know. And I'm collaborating with the GAR people to get more data and try to understand. And there was also a paper more recently in Amphioxus. So Amphioxus, if you look at the phylogeny, is the outgroup to the vertebrate genome duplications. And what we find here, we cannot really distinguish. So this is a complicated figure, but what's important that when you have homologs, you can compare the expression in the tissue in the Amphioxus to the zebrafish. And what you often have is a kind of specialization. So it's not really a subfunctionalization in the cartoonish way, but more that one of the, two zebrafish copies is broadly expressed in more or less the same place as the Amphioxus, and the other is much more narrowly expressed. And this is something we see a lot. We also saw in the human mouse data, although we didn't have an outgroup, is that we have a lot of cases where when there's two duplicates, one is lower expressed and more specialized, and the other is higher expressed and more broadly expressed. But how this relates to subfunctionalization and neofunctionalization, we don't yet know. So this is something, this third hour, I'm sorry, is full of question marks. We don't have the full answer to the ortholog conjecture. We don't have the full answer to neo and subfunctionalization. That's research for you. But I think we are refining our view and seeing that things are more subtle than the cartoon images we had 10 years ago. So if I want to keep a few minutes for questions, I will go to conclusion slides. So on evolution after genome duplication, there is evidence for both sub and neofunctionalization. And one of the things I think that people would like to do now is to understand better, clearly both happen. When do they happen? Is it because neofunctionalization happens more short-term and subfunctionalization long-term? Is it because different categories of genes are kept? Is it because some are at the expression level and some at the protein sequence level? I didn't show you, but there's starting to be some evidence for selection on the protein sequences. Also, many genes do not diverge in expression. I didn't show this too much in the slides because it's kind of boring pictures, but in fact, these slides are only a part of the picture. Here you have the genes diverge between the copies, but salmonids have many more genes than this. They have about 40,000 because they had several genome duplications. And most of them are actually all the same. And we see this also, for example, in Xenopus. And we're studying a lot these questions on expression because it's I would not say easy, but feasible, tractable, to get expression in different species across the whole genome and compare it. Other features of function are much harder. So they're harder to get large scale. And when we get them large scale, they're harder to compare. So we have limitations both on the experiment side. It can be very difficult to do, it's almost impossible to do systematic knockout in any species which is not yeast or E. coli. It's very difficult to do systematic protein-protein interactions. It's very difficult to do systematic, and most then features of function are actually specific of subcategories of genes. So if you have enzymes, you want to study the catalysis. If you have receptors, you want to study their ligands. If you have transcription factors, you want to study the genes they regulate, but then this becomes much harder to put all together. And bioinformatically, as soon as you go outside of RNA-seq, which already has biases, then it becomes even worse, like what I showed you for the gene ontology, complicates also all the other things, whether it's knockout phenotypes, whether it's protein-protein interactions, whether it's enzyme activity, all these things become very complicated to compare. So I'm not saying we won't manage, but it's clear that we start with expression because it was tractable. So in conclusion of this morning, thank you for listening first. I see that some of you are still here. So I hope I showed you that gene and genome duplication are quite common. And so we should take them into account in comparative genomics. I think I showed you that the evolution of function is hard to study. I don't want to discourage you. It also means it's interesting. Challenges make for interesting research. So far, as I was saying, expression is the best studied proxy for functional evolution. It doesn't mean it's the best. And maybe in 10 years, we'll look back and say everything we did on expression was really crappy and lucky now we have a better way to measure function. But that's what we can do now. Okay, so we're like the tale of the drunk person who is looking for their keys under the street light, not because they dropped the keys under the street light, but because that's where there's light to look for keys. And finally, I hope I showed you that curated databases are useful, and to study function, we really need this curation and these structured databases. Thank you for your attention. I will now look at the questions. So questions. Are pseudogenes included in subfunctionalization? No, pseudogenes are included in loss of the gene. So a pseudogene is considered a step between towards losing the gene in most cases. So if you have 2 copies of the gene, I showed you only the scenarios where you keep the 2 copies. There's actually another scenario, which was the most common one. You have 2 genes and now you go back to 1 gene. You lose, and that's that exponential decay we had at the beginning of the morning. And now how do you lose genes? It's very rare that molecular scissors come and cut out exactly one of the 2 copies. What happens is that you have mutations which little by little deactivate this gene so that it's not expressed, it's expressed but it's not translated, it's translated but it's not folding, So you get a pseudogene. Pseudogenes can be transcribed, okay, sometimes. And the pseudogenes are basically duplicate genes being lost. And actually this brings back to the question someone had earlier this morning. If duplication is frequent, we should see a lot of duplicate genes in the population. And I spoke of copy number variants. Actually, the pseudogenes, and there's a bit less pseudogenes than genes in the human genome, but close. So it's like 19,000 to 17,000. Sorry, I don't remember the exact numbers. The pseudogenes are all evidence for duplication, which happened not so long ago and is being lost. Because once a pseudogene has no function at all, is not under any purifying selection, then it'll be a target for mutations which will occur and get fixed by drift without any selection to prevent them from getting fixed. And so pretty fast, you will lose them. So you cannot compare autologous non-functional sequences between a human and a mouse. That's too far away because after 80 million years on each side, so 160 million years total evolutionary time, mutations scrambled things so much that you lost everything. And you should know that in most, at least eukaryotic genomes that I know, small deletions are more frequent than small insertions. So if you have a piece of sequence which is useless, you basically lose it little by little just by deletions. if you wonder why our genome isn't then much smaller, it's because we also have transposons who come and increase the size of our genome, but that's a different discussion. Going to the questions. Okay. Does co-expression of two genes speak for subfunctionalization? Co-expression of two homologs or paralogs, or co-expression of two unrelated genes? Paralogs. So if two paralogs are co-expressed, that says that they share function. So it's from an expression point of view, it would favor just increase in quantity. Now I know anecdotal stories where there's shared expression with changes of function, but it's hard to study large scale. But for example, estrogen receptor in vertebrates, there are two genes, estrogen receptor alpha and beta, and they form a heterodimer. And they express together, and you need both paralogs to form the functional heterodimer in some circumstances. Sometimes you need the homodimer actually, and sometimes the heterodimer. So the heterodimer has a new function. And you have various cases like this. The most well-known is hemoglobin. Hemoglobin is an assemblage of paralogs which are expressed together, but have different subfunctions. So yes, this can happen, but it's hard to characterize on a large scale because you can look in detail at one transcription factor or one globin, but to characterize this large scale is difficult. Would you consider duplication somewhat useful because we can compare them to an outgroup? Useful for what? Because it depends. Useful is useful for something. I like duplication because I studied them. So they're very useful to my career. I'm not sure what the question is. If this is the question, I know some papers which use duplications as an additional phylogenetic information. So they improve the species phylogeny by the dating of the duplication. So if that's the question, I can give you some links to that. Yeah, so I can give you there are some methods which exist, and probably Christophe Desimo knows also some others which use the order of duplications in the phylogeny as an additional evidence for species phylogeny. Additional question, as a rough measure, can you use the number of present absent cows in Bgee? Use it for what? So use it as a measure of tissue specificity? Yes, you can, if that's the question, yes. And in our benchmark for tissue specificity measures, we showed this works not as well as a quantitative measure, but not too bad. And actually, we're implementing this into Bgee, but it's not yet on the website because before we put something on the website, we have to verify it 1 million times. So yeah. Is there a cause or biases of genomic analysis? Seems like a vast and interesting subject. The thing is, once you look into it, it's interesting, but if we put this as a title, no one will come. So it's an interesting suggestion. I don't know if Patricia Palagi is here or not right now, but yeah, she's here.
I am.
She's coordinating the training at SIB, so we can take note of the question. Like, you know, when genomic methods don't work, it would be fun to teach. I don't know if a lot of students would come.